I'm Rick Lifton, the president of the Rockefeller University, and I am so thrilled to welcome all of you to the 67th Convocation of Rockefeller University. It is just great to see so many of you gathered here in Caspery Auditorium to share this important milestone. If this is your first time at Rockefeller, by now I suspect you've discovered that we're unlike most other universities you know. We don't have undergraduates, we don't have a law school, an English department, or sports team, though I note that pickleball has really taken over the athletic environment here in the last year. But what we do have is a singular focus on pioneering biomedical research. Our clarity of mission, science for the benefit of humanity, has been our North Star, our guiding principle since our founding by John D. Rockefeller in 1901. A core belief of ours has been that understanding the detailed biology underlying life's intricate processes will provide the best opportunities to ultimately prevent, diagnose, and treat human disease. And second, that the very best scientists are typically underfunded, and that providing exceptional scientists, exceptional resources, and an encouraging and engaging environment will lead to greater impact than spreading the same resources more thinly across 5 or 10-fold more faculty doing less consequential research. We strive to recruit the most exceptional scientists from around the world and empower them to tackle bold and consequential questions. We're supported by a board of trustees that champions innovation as passionately as our scientists do, and most importantly for today, an exceptional community of doctoral students whose creativity and dedication drive our discoveries forward. The results of this approach speak volumes. Though we have only had about 260 professors in our history, 26 of them have received Nobel Prizes in medicine or chemistry, and another 26 have received Lasker Awards. This group includes 2 Nobel Prizes and 3 Lasker Awards in the last 9 years. And most recently, Svetlana Moisov, an alumna of our Ph.D. program, received the 2024 Lasker Award last fall for her discovery of the peptide hormone GLP-1, which I suspect a few of you may have heard of. This has led to highly effective treatments for diabetes, obesity, and a striking reduction in the risk of obesity's consequences, including cardiovascular disease, kidney failure, a number of obesity-related cancers, and likely a range of obsessive or addictive behaviors, and as yet undefined opportunities in a range of neurodegenerative disorders. Since 1959, when the first 5 doctorates were conferred from our PhD program, our alumni have gone on to highly impactful careers at the world's leading universities in the biotechnology and pharmaceutical industries and in other fields that require strong analytic thinking. To cite one very recent example, just 2 weeks ago, a 2004 graduate of our program, Kiran Musarunu, now on the faculty at University of Pennsylvania, published a remarkable story. An infant was born with a mutation that resulted in an inability to make a chemical called orotic acid, and this results in crises of accumulation of ammonia that is normally cleared from the blood but is not done in the absence of this enzyme. And this inevitably causes severe brain damage in a matter of months after birth. Once the specific mutation causing this program was identified using high-throughput DNA sequencing, within several months, a bespoke genetic treatment using DNA base editing technology to selectively correct the single mutation in the human genome was developed, tested in animals, approved for use, and delivered to the child's liver in a lipid nanoparticle by intravenous infusion, with the base editor finding the specific mutation and changing that one base back to the normal sequence, which miraculously restored the ability to produce orotic acid and has prevented elevations in blood ammonia levels and allowed increased dietary protein intake. This treatment is expected to find a lifelong prevention of disease in this child. This was unimaginable 15 years ago, and when I started and many of my faculty colleagues started their careers, couldn't even be contemplated, as we just didn't have any idea how to achieve such a remarkable outcome. Today, we are immensely proud and excited to be conferring the Ph.D. degree upon 43 graduating students. This has been a truly exceptional group. They've been integral members of our community of scholars who, through their inspiration and hard work, have become outstanding scientists in their own right. Convocation is also a moment to honor individuals whose efforts exemplify the power of science to advance humanity. We recognize these individuals not only for their achievements, but to inspire our entire community as exemplars of what is possible when one dedicates a life to scientific discovery, the pursuit of truth, and service to humanity. They demonstrate a standard to which we all can aspire. This afternoon, we will confer the Doctor of Science honoris causa upon 4 remarkable individuals: Quarraisha Abdool Karim, Salim Abdool Karim, Marlene Hess, and Michael Young. Today's gathering is possible thanks to the enduring leadership of the Rockefeller family, and in particular, to the vision and dedication of David Rockefeller. David understood the vital role of educating future scientific leaders who will build upon what is known and will make discoveries that will far surpass our prior understandings and will eventually pass on their knowledge and lessons to their successors. This unbroken chain of discovery has been remarkably powerful in advancing human health, which in 1901, when Rockefeller Research Institute was founded, life expectancy in the United States was 47 years. Over the last 125 years, life expectancy has increased to nearly or more than 80 years in many countries worldwide. David Rockefeller was deeply involved in shaping our graduate program, and it is with great pride that this program bears his name. Our university thrives without formal departments and subject matter boundaries. This allows our students to be immersed in a uniquely collaborative and flexible environment. They learn from incredible scientists working in diverse areas with very distinct technologies and approaches. They also learn from one another and graduate not only with their own notable scientific accomplishments, but also with broad exposure to emerging areas of impact in the biomedical landscape. The goal of our Ph.D. program is to promote in our students the curiosity and scholarship to identify important problems, to help them develop the knowledge needed to develop experimental approaches, to test ideas designed to solve these problems, as well as the technical and analytical skills that are critical to completion of their Ph.D. thesis. We also hope we develop in our graduates the love of pursuit of new truths about life on this planet, and the remarkable thrill of discovering something new about the secrets of life, made deeper that the knowledge that we collectively are adding to the remarkable body of knowledge about life and its impact on the betterment of humanity. We trust that our graduates will go on to use these skills well. The importance of ensuring an enduring pipeline of well-trained biomedical scientists has, of course, been on full display during the Ph.D. careers of this year's graduates. All of this year's graduates were here when COVID exploded here in New York City in March of 2020. Indeed, right here at two major hospitals just across the street and right next door at the corner of 68th Street and York Avenue. The resulting pandemic was tremendously disruptive to our students' lives and their ongoing research, and yet these students were not only resilient, they rose to the challenge, in many cases continuing work on their projects and contributing to the biology, diagnosis, and treatment of COVID-19, all while adapting to the challenges of the workplace to prevent infection among our coworkers. This year's students have succeeded greatly, working collaboratively in highly diverse areas of biomedicine, and from this experience, they will carry forward a more immediate understanding of the critical importance and urgent value of science to humanity than most who came before them and who will follow this year's graduates. The application of lessons from fundamental science to the pandemic proved crucial to the unprecedented speed of the development of highly effective vaccines and therapeutics that prevented severe disease and death from COVID, and the development of a detailed understanding of the evolution of this particular virus and the factors that predispose to severe illness resulting from infection. It was a spectacular demonstration of the power of science that saved millions of lives worldwide. Nonetheless, this success has been tempered by the knowledge that so many people in the United States and around the world died needlessly from widespread misinformation about the virus, the vaccine, and effective therapeutics, as well as the lack of universal access to these vaccines and beneficial therapies. For all these reasons, we hope and expect that this year's graduates will use their hard-won skills as voices in our society, promoting the importance of the pursuit of truth and facts, the central tenet of the scientific method. Although the skills of our graduates have been honed working in academic biomedical science, they will be useful in any enterprise and any forum in which an inquisitive mind and exceptional analytical skills are of value. We trust that our graduating students have learned that the curiosity, creativity, and passion needed to learn something new about how life works is both a treasured talent and that success in making such discoveries is endlessly thrilling, and that these new insights will have dramatic impact on health and disease. Despite these amazing advances and opportunities, this year's graduates are nonetheless receiving their PhDs in unsettled times, with unprecedented challenges to science and to many research institutions across the nation. We are now facing the potential of extreme cuts to federal funding for biomedical research, which has been the driver for scientific discovery that has made the United States the world leader in science and technology. Basic research in universities has provided the critical foundation upon which the biotechnology industry was originated and built upon uniquely in the United States. This ecosystem supports more than 3 million jobs in the U.S. and is a major contributor to the U.S. economy. And beyond this economic impact, discoveries in universities are the substrates upon which new medicines have been built and have transformed millions of lives. These remarkable advances make it hard to fathom why the government would be actively seeking to curtail our nation's progress in science and cede clear U.S. leadership in biomedicine to the rest of the world. Part of this comes from politicization of science, with huge amounts of misinformation coming from both the administration and social media. An opinion piece published just yesterday referring to these cuts in science and technology said that we are witnessing a superpower suicide. It's hard to think otherwise. These cuts extend as well to the administration's elimination of many USAID programs in Africa and elsewhere that have been critical to the treatment or prevention of HIV infections, tuberculosis, and malaria, to cite just a few, including as well malnutrition. This will undoubtedly cause much suffering, not just abroad, but ultimately affect us here as well. And recently, the federal government has also issued edicts seeking to slow or prevent issuing of new visas for international students to enroll at U.S. universities, including ours. Here at Rockefeller, we proudly seek the best students and faculty from around the world, with about 40% of our students and faculty born outside the U.S. This has been a continuous source of strength for science and the U.S. economy, and we remain fully committed to recruiting the most promising people we can find wherever they are born. Despite recognizing the fraught times we live in, one can't be a scientist without an optimistic worldview. It seems requires a fair amount of hubris to assume that you can discover something new about how life works and its potential application to human health, and yet we continually produce such new knowledge that does have impact. The advances in science and technology are truly extraordinary, and we must keep our optimism that the U.S. will not irreversibly go down a nihilistic path. Science remains a remarkable pursuit with a beautiful goal. This new knowledge adds to our prior understandings, and these discoveries are of great benefit. The advances of the last 50 years have been truly extraordinary and were unimaginable. I'm certain discoveries in the next 50 years during the careers of this year's graduates will similarly dwarf what has come before. And I know that students graduating today will be important contributors to these future advances, and I wish them all every success. The world is counting on you. Congratulations.
Good afternoon and welcome. My name is Tim Stearns. I have the privilege of serving as the Dean of Graduate and Postgraduate Studies here at Rockefeller University. It is our tradition at Rockefeller to focus this convocation ceremony on the graduate. Mr. President, Vice Chairman Robert Steele, graduates, Rockefeller community, honored guests. I now give my formal assurance that these graduates have satisfied their obligations and are eligible to be conferred the degree of Doctor of Philosophy.
All right. Good afternoon, everybody. Parents, family members, friends, and significant others. Thank you for coming here to celebrate our graduates, and it's a privilege for me to present the first of the graduates graduating today, Christian Baca. So Christian graduated from University of California, Irvine in 2019 and joined the Transinstitutional program in chemical biology. He rotated in my lab in the most unusual circumstances, because he started in February 2020, shortly after campus or before campus closed during the first months of the SARS-CoV-2 pandemic, and finished the rotation when the labs reopened with social distancing restrictions in June of 2020. My lab uses molecular genetics tools to study bacterial immunity, and Christian's previous laboratory experience was in peptide synthesis. Therefore, not only the work environment was difficult for Christian, but also the research we performed in the lab must have been challenging for him. Nevertheless, he decided to join us, and in spite of limited in-person interactions that we had, I made the excellent decision to accept him into the lab. Bacteria, as much as humans, are also infected with viruses, which are known as phages. So Christian worked on the characterization of immune effectors called CARF, that participate in the type 3 CRISPR-Cas immune response of bacteria. In collaboration with the Dean Shaw Patel's lab at Memorial Sloan Kettering Cancer Center across the street, he characterized 3 of these effectors. While he was not very creative in naming them, because he named them CAM1, CAD1, CAT1, which are almost impossible to distinguish between these names, his work, nevertheless, uncovered an astonishing diversity of enzymatic activities and structures for these proteins. And he landed the perfect hat trick in publishing, because each of these studies were published in Nature, Cell, and Science, something unprecedented in my lab. So Christian's work with really high intensity, and it has been a pleasure and an honor to be his mentor. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Christian Baca.
Nicholas Blobel joined my lab with the goal of identifying endogenous sources of DNA damage that, when left unrepaired, lead to cancer. He pursued two distinct but equally challenging projects, and to be brief, I'll describe just one of them, the one that focused on uncovering DNA-damaging metabolites in the keratinocytes, cells that line our mouth. For this project, Nicholas used keratinocytes that lacked Fanconi anemia DNA repair pathway. This pathway is critical for removing DNA interstrand crosslinks, a very deleterious kind of damage that inhibits replication and transcription. Deficiency of this pathway leads to inevitable oral and esophageal cancer in Fanconi anemia patients. This project proved to be technically demanding, as endogenous DNA damage accumulates gradually, making it difficult to detect measurable changes, both ex vivo and in vivo. However, Nikki's tenacity, perseverance, and creativity in developing innovative approaches were key to overcoming these challenges. These experiences not only honed his ability to think independently and solve complex problems, but also instilled the resilience and boldness necessary to achieve his goal of becoming a physician scientist and a principal investigator. To uncover the sources of DNA damage in oral keratinocytes, Nicholas conducted a series of CRISPR editing-based screens and identified endogenous aldehydes, super-reactive molecules that can create interstrand crosslinks, as key culprits. Specifically, he found that a deficiency in ADH5, the primary enzyme responsible for detoxifying formaldehyde, a metabolic aldehyde byproduct produced at high levels in our cells, leads to poor survival of Fanconi-anemia-deficient keratinocytes. In a mouse model of Fanconi anemia, ADH5 loss results in oral dysplasia, further supporting its role in preventing DNA damage-driven malignancies. This leads us to believe that formaldehyde, especially in cells that are not protected by the Fanconi anemia DNA repair pathway, creates mutations that lead to squamous cell carcinomas of the oral cavity and esophagus. Beyond his research, Nicky's unwavering commitment to becoming a physician scientist extended beyond the lab. He remained actively engaged in patient care during his Ph.D., where he worked to improve healthcare access to underserved patients. And during the COVID-19 pandemic, he further demonstrated his dedication by volunteering for up to 10-hour shifts as a vaccinator. His seamless integration of research and patient care underscore his exceptional potential as a future physician scientist. Nicky is now completing medical school and is planning on matching and research track internal medicine residency. Mr. President, Mr. Vice Chairman, and our honored guests, it's my pleasure to present to you Nicholas Blobel.
Congratulations. Hold your photograph. Hold on. Thank you very much.
New technology is exciting. It can provide new ways to answer long-standing questions, and it can even let us ask new questions about how biology works. But it comes with challenges. One is, of course, all the troubleshooting needed to get the new method to work. But even once that's done, and those challenges are surmounted, a big one remains. How do you analyze a new type of experimental data to draw biological conclusions? We faced this challenge in my lab as we sought to apply RIC-seq, a new method for measuring DNA contacts within cells by sequencing the DNA fragments produced by X-ray radiation. Our ability to interpret the data was limited to imperfect and overly simplistic models of chromatin used to create a simple framework for interpreting the data. In her thesis work, Ariana Brenner-Clerkin came in and worked first as part of a team and then on her own to build and validate a nanoscale simulation of chromatin structures with different basic properties. This was much more realistic than what we had done before, and she used the results of these simulations to train a convolutional neural net to extract biologically meaningful chromatin properties like the spacing of nucleosomes, the basic building blocks of chromatin, the wrapping of DNA around these building blocks, and other interaction parameters that we are now using to analyze RIC-seq data so that we can understand how proteins mutated in cancer shape DNA folding. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Ariana Brenner-Clerkin.
Hi, all, and welcome. It's my privilege to introduce Josh Brewer on behalf of myself and his co-advisor, Seth Dars. So as Seth puts it, Josh is a bit of a mystery. He was born in the UK, raised in Arizona, studied in Egypt, France, and China before graduating with honors in politics from Pomona. So he's worked in journalism, he's published in Middle Eastern affairs, and then he later pivoted to protein biochemistry and X-ray crystallography at Columbia. So by the time Josh was interviewing for graduate school in 2017, Seth was determined to recruit this person, and he did get an offer from Rockefeller, and thank God that he did, because Josh's work has rewritten the transcription cycle paradigm. In Seth's lab, he's discovered a new step, post-termination ejection, and that made us all have to update our slides and probably have to update the textbooks as well. And in my lab, he revealed the molecular intricacies of the step that rifampicin targets in the transcription cycle. And these are critical insights in the way we treat TB. So across both projects, Josh has shown exceptional scholarship in writing and an unmatched command, unmatched to mine and Seth's in 50 years of transcriptional literature. He's a deep thinker, and now I find myself turning to him for advice. And that's a sure sign that the student has outgrown the mentors. He's unapologetically, a fun fact, he's unapologetically committed to cinnamon raisin bagels with jalapeno cream cheese. So he's been banned from Bagel Works. And he brought the house down at a conference performing Hey Ya at karaoke. And then he actually got a job offer. Someone came up and asked me, who is that? And he got a job offer. So I just want to end by saying, he's brilliant, he's thoughtful, full of range, from Jeeves and Wooster to Star Trek to ABBA to politics. He's one of a kind. I'm thrilled he stayed on for a year as a postdoc with me to help launch some in situ structural studies we're doing in TB. I'm equally thrilled and sad, too, to say goodbye to him on his next chapter as an equity research consultant. Mr. President, Mr. Vice Chair, and honored guests, it's my pleasure to present to you Joshua Brewer.
Congratulations. Well done.
It is my great pleasure to introduce Chloe Burnside. Following her undergraduate studies at Imperial College London and the ETH Zurich in Switzerland, Chloe joined first the tri-institutional program in chemical biology and in 2019 our lab. For her thesis work, Chloe pioneered a new research direction in the lab to study mitochondria, the powerhouses of cells. With their own genetic code and genetic information, these organelles require their own ribosomes for protein synthesis to enable energy production. Yet how these mitochondrial ribosomes are themselves assembled had remained unknown, and Chloe decided to pursue this problem using yeast as a model system. She set up an investigation into how the final architecture of mitochondrial ribosomes, which are large RNA protein complexes, is related to the mechanisms by which these molecular machines are assembled. Her pioneering work has set the stage for how we think about the evolution of RNA processing and ribosome assembly in mitochondria across species. These principles have fundamentally changed our understanding of these pathways in ways that were completely unexpected. It has been a privilege not only to see Chloe grow as a scientist in the lab, but also to see her communicate science in the wider community through Rock.edu, where she's been teaching and mentoring the next generation of scientists. Looking into the future, I cannot wait to see what she will achieve in her next endeavor. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Chloe Burnside.
Thank you.
Gabriela Chua never ceases to break new ground. Growing up in the Bay Area as a gifted multi-sport athlete, she came across the country to attend Bates College, where she completed an exceptional undergraduate thesis project that led to a first author publication and the first ever patent at Bates. Gabriela then brought her talents to New York, attending the tri-institutional Ph.D. program in chemical biology. Trained as a biochemist, Gabriela became interested about the potential of using physical tools to learn biology and joined my lab to conduct her PhD research. With a deep-rooted interest in epigenetics, Gabriela first set out to study the methyl CpG binding protein 2, a master regulator of gene expression in the brain, whose mutations cause a devastating neurodevelopmental disease called Rett syndrome. Supported by an NHF31 award, she performed decisive experiments that uncovered for the first time the dynamic behavior of this mysterious protein on chromatin. After this feat that culminated in a landmark paper, Gabriela once again looked for a new challenge, this time turning her attention to the fundamental process of DNA replication. In collaboration with the Michael O'Donnell's lab at Rockefeller, where she will continue with her postdoctoral research, Gabriela reconstituted and visualized this process with unprecedented resolution. This allowed her to discover that a key enzyme known as the clamp loader does not always turn over after loading its substrate onto DNA, but rather remains associated with the replication machinery to enhance its performance. This finding challenges the textbook model and profoundly changes our view of genetic inheritance and genome maintenance. Beyond all these research achievements, Gabriela has also put in extraordinary efforts into community service and science outreach. Whether it's Asian Perspectives on the Hidden Curriculum discussion series that she launched here at Rockefeller, or the Weill Cornell High School Catalyst program where she served as co-president, Gabriela always makes herself available to offer help, advice, and camaraderie. I see from her, and I see from many others in this graduating class, the essential qualities needed for a leader in sciences and society: innovative, resilient, and compassionate, which make me firmly optimistic, even in these precarious times, that our future is bright. Mr. President, Mr. Vice Chairman, and honored guests, I proudly present to you Gabriela Chua. Congratulations. Congratulations.
All right. Born in Sao Paulo, raised between continents, Philippe's path to Rockefeller was anything but linear. He's been a punk rocker, a synthetic organic chemist at Johns Hopkins, and today we celebrate him as a molecular microbiologist whose thesis reshaped how we understand one of TB's most essential systems. Philippe set out to explore a remarkable protease system called CLIP. This protease is essential in TB and a high-priority drug target. But despite its importance, no one really understood why it was essential, and Philippe suspected that the published explanations were likely wrong. Through a series of elegant in vivo genetic experiments, he uncovered that the real story was far more complex and far more interesting than we had appreciated. Philippe's findings reshaped how we think about CLIP. Perhaps even more importantly, his work showed that ongoing drug discovery efforts were headed in the wrong direction and helped steer them back on course. Not content with this, Philippe continued to dig into the question of why CLIP is essential, work that's now being carried forward by another student in the lab. Philippe has since returned to the MD side of his MD Ph.D. journey, and we send him off with admiration, gratitude, and confidence in the impact he'll continue to have, whether it be in the clinic, in the lab, or wherever he chooses to go next. Mr. President, Mr. Vice Chairman, and honored guest, it is my pleasure to present to you Philippe D'Andrea, Dr. Philippe D'Andrea. Thank you. Okay, so Catherine grew up on Long Island and joined the Rockefeller PhD program after completing her undergraduate studies at Stony Brook University. She stood out even then for her drive, her deep curiosity, and her unshakable commitment to understanding the biology of tuberculosis. Catherine's thesis work focused on a fascinating and pressing challenge in TB. How does drug resistance alter the physiology of the bacterium? And do these changes reveal new weak points, new Achilles heels that we might exploit? We've long known that while drug resistance gives TB a clear advantage in the presence of antibiotics, it often comes with a fitness cost when the antibiotic isn't around. Catherine hypothesized that by identifying which genes these resistant strains rely on more or less, we could begin to understand the basis of that fitness cost. And she was right. Catherine's work, in collaboration with our colleagues Liz Campbell and Maddie Delbo, uncovered a previously unrecognized class of compensatory mutations in NUSG, a gene encoding a transcriptional regulator. She showed that these mutations help resistant TB strains overcome transcriptional defects caused by one of the most common drug resistance mutations. In doing so, she not only taught us why these drug-resistant bacteria have a fitness cost, but how we might exacerbate that cost to try to overcome resistance. Catherine's thesis is a tour de force in functional genomics, mechanistic microbiology, and translational insight. Catherine now moves on to a postdoctoral fellowship with Sabina Ehrt and Dirk Schnappinger at Weill Cornell, where I know she'll continue to do exceptional work. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Dr. Catherine Eckert.
Congratulations.
Thank you. Thank you. Okay. Hi again. It is really my honor to introduce Ruby Froome on behalf of her co-advisors, Jeremy Rock and Seth Dars. Ruby came to Rockefeller already a star, summa cum laude graduate of Williams College and an accomplished musician. So in 2018, she wrote to Seth and she said, I've always been interested in bacterial pathogenesis and I'm drawn to mechanistic studies. So therefore she joined my lab, Jeremy's lab, and Seth's lab, and she lived up to those words. She charted her own course at developing a method she calls cell-free genomics. And in doing so, she made real what had always been a dream in the field for a while, genomics in a test tube system. Along the way, she discovered regulatory features and key TB transcription factors, and she's now carved a place out for herself in the world of transcriptional biology and in genomics. I don't say this lightly. Her work is both pioneering and profound. Jeremy says it best, that Ruby is one of those rare PhD students who can absorb ideas and truly chart her own path, and she did. But if I had to single out a single quality of Ruby, one that shines the brightest, it's her intuitive ability to really draw connections, really critical aspect in science. Her PhD work revealed both independence and a mind that was truly special. She's also incredibly kind and a committed mentor. She's committed to service, and she guided a Yalo scholar, taking quite a bit of time away from her work. But she's also been awarded many honors for some of this, including the Women in Science Graduate Fellowship, and she was elected chair of the 2025 GRS in TB drug discovery. So her fun fact is that you never want to follow her after karaoke. She's a phenomenal singer and a pianist, and you really should look her up at on YouTube. I'm really, right after this, do that. So I'm not exaggerating. You'll see I'm not exaggerating. So Ruby's sincerity, her brilliance, and her generosity has really made Rockefeller better. She was quickly recruited before she even graduated to Regeneron, and I miss her immensely. So Mr. President, Mr. Vice Chairman, and honor guests, it's my pleasure to introduce you to Ruby Froome. Present.
It's my honor to present Audrey Goldfarb, reading the comments of her thesis advisor, Titia DeLongo, who could not be here today. Audrey came to the Rockefeller Graduate Program in the fall of 2019. After rotating with the Smargazeska and Darnell labs, she had planned her third rotation in my lab when the pandemic hit. It was late March 2020, and I was very concerned about Audrey and other first-year students sitting alone in their very small rooms without friends or support. Audrey initially volunteered to help up Darnell's lab with setting up the PCR COVID-19 saliva test that later helped prevent the spread of infections in the university community. Audrey's delayed rotation started much later in 2020 when we were returning to a semi-functional state, but still with taped arrows on the floors directing traffic in the lab, signs indicating the maximum number of occupants per room, and social distancing rules. Audrey worked under the guidance of Nanda Sasi, a postdoc who proved to be an exceptional teacher and patient mentor. The goal was to understand how the telomeric protein TRF2 forms T-loops and how T-loops prevent the DNA damage response, key questions in telomere biology. Audrey and Nanda made excellent progress on this very challenging project, and their paper received positive reviews. 2 research assistants in the lab have been hard at work addressing the reviewers' comments, and we're optimistic we will get this excellent work published soon. In addition to her work in the lab, Audrey contributed to the Rockefeller community by chairing the Anderson Center for Cancer Research Symposium Committee, working with RISI, contributing to the LGBTQ community, and single-handedly reviving the moribund Natural Selections publication that serves to provide some lighter fare to our community. She is now a postdoctoral fellow at the University of Pennsylvania. Mr. President, Mr. Vice Chairman, and honored guests, it's my pleasure to present to you Audrey Goldfarb.
Congratulations, Audrey. Well done.
Congratulations.
It's a great pleasure It's a great pleasure to present to you Mason Hargrave. Through the years, my lab has attracted a considerable number of brilliant young scientists with, more often than not, interesting personalities. And Mason has distinguished himself in both of these areas. When he interviewed for the graduate program, the paper said that he was from rural California and that he had majored in physics at UC Santa Cruz. So I was expecting your standard, disheveled, unkempt nerd. Then at the door of my office, there's this tall dude with booming voice, dressed to the teeth in, I think, a houndstooth vest, a tweed vest, ankle-length pants, closely cropped hair, looking far more like a banker than what I expected a physicist to look like. He would reinvent his appearance many times, through his Ph.D., okay, throwing us off guard on a number of occasions. So after a few rotation projects, Mason decided to join both my lab and Connor Lifton's lab at Cornell to develop biometric markers for depression. But he quickly learned that in the clinical environment, changing anything, even something as simple as using a good microphone to make interviews, was fiendishly difficult. And this led him to what was, in fact, his thesis. Okay, a central quandary for data-hungry AI methods is the scarcity of information and of data we have on changing interventional policies, particularly in changing what we call dynamic treatment regimes. We have a lot of clinical data on tests, but we don't have a lot of clinical data on making decisions and changing those decisions. As a result, many practitioners have turned to a technique used, in the industry of simulating more data, using AI to take the data you have and simulating more. This is somewhat dubious practice that has, worked very well in areas where you have a lot of data. But in collaboration with Professor Logan Grosnick at Cornell, then Mason developed a benchmarking tool, okay, to assess the effectiveness of these off-policy and off-regime methodologies. And he proved without a shadow of doubt that there is simply no shortcut in a data-hungry, in a data-deprived field, okay, other than actually getting the data, as dangerous and painful as it is. So in a field that's full of vapid promises, Mason followed the science with rigor, okay, and led us to a scientifically rigorous result. I do have to report that Mason did not succeed at one of the tasks he set for himself. You see, at the Faculty and Students Club, my laboratory holds the record for the most bottles of champagne consumed after a thesis defense celebration. And Mason valiantly tried to up that record, and he was unable to. So Mason has now embarked in a new career as a consultant with BCG. And as much as we will miss him in academia, okay, we wish him the very best in his endeavor. So, Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to introduce you to Mason Hargrave.
It is my great pleasure to introduce Nathan Harper. When Nathan joined the lab in 2020 to visualize the biogenesis pathway through which human mitochondria assemble their ribosomes, this seemed like a near impossible feat. In retrospect, it would seem naive at best to let anyone take on a project like this single-handedly. However, Nathan was determined and did just that. Through a series of experiments, Nathan's work described the early stages during which mitoribosome assembly is catalyzed by a series of enzymes. This work not only illustrated the precise level of control that is achieved by coupling irreversible steps in an assembly pipeline with conformational and compositional changes within maturing mitoribosomal particles, but also highlighted how important functional centers of these particles are crafted in a staged manner. With his subsequent exploration of the same pathway in bacteria has highlighted both similarities and differences that exist between human mitochondria and their bacterial ancestors. Altogether, Nathan's work has allowed us to appreciate the evolution of macromolecular complexity and how it is intimately linked to the evolution of assembly pathways. Nathan is a force to be reckoned with, as someone whose talent is initially obscured by his understated nature, but unleashed at the right moment. Having seen how he has revealed much of what we now know about the assembly of mitoribosomal subunits in human cells, I have no doubt that his next steps as a postdoc in the UK will once again bring his talents to light. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Nathan Harper.
Congratulations. Well done. Good job.
Thanks.
It's my honor to present David Shea, giving the remarks for his thesis advisor, Sean Brady. David was a member of the tri-institutional program in chemical biology during his graduate studies. As an undergraduate at National Taiwan University, he conducted research on single-molecule microscopy in real time. When he joined my group, he became very interested in mastering the nuances of high-resolution liquid chromatography mass spectrometry and using his newfound expertise to investigate the diverse small molecules that are produced by the gut microbiome. David is exceptionally tech-savvy by nature. He designed an elegant series of mass spectrometry experiments to first catalog gut bacteria that produce molecules related to human-derived metabolites, and then to determine whether any bacteria produce metabolites that are exact copies of native human signaling compounds. His work led to the discovery of a common group of gut bacteria that produce a molecule identical to a human-derived signaling compound known to regulate food intake after exercise, as well as influence long-term weight gain. This represents a rare example of a commensal bacterial species producing a perfect molecular mimic of a human signal. While further research is needed to understand the long-term effects of colonization by these bacteria, David's analyses of large publicly available data sets suggest that young children with high levels of these bacteria in their gut microbiome tend to gain less weight over time. David is now applying his deep expertise in mass spectrometry at a startup company here in New York City. David is not only a great scientist, but he is also a great person. In science, where setbacks are inevitable and most experiments fail, he always sees the positive side of the challenge and remains optimistic. He will be dearly missed by his lab mates and those who have had the privilege of working alongside him. Mr. President, Mr. Vice Chairman, and honored guests, it's my pleasure to present to you David Shea.
Thank you, Tim.
Congratulations.
Haejin Kim. I'm delighted to introduce Haejin. Haejin is originally from New Jersey and attended Rutgers University, where she worked in the lab of Richard Ebright studying bacterial transcription and graduated summa cum laude. In 2018, she came to the Transinstitutional MD Ph.D. program and joined my lab in the summer of 2020. And again, another story of COVID. These were difficult times with social distancing and lab work shifts in place. Therefore, Haejin has to be very self-sufficient to start her thesis, not only regarding receiving help to learn new techniques, but also in terms of the projects he decided to work on. And she did exceptionally well, and I consider Haejin one of the most independent students that graduated from my lab. As I mentioned before, my lab works on how bacteria use CRISPR systems to defend themselves from viral infection. She did a thorough reading of the CRISPR literature of the time and decided to characterize a mutant version of the Cas9 nuclease that we identified in 2015 as having enhanced immunity properties. We had no idea how this worked, and I thought this was a very difficult project. But Heijin performed elegant experiments that elucidated unexpected aspects of Cas9 function and regulation. I had a great time working with Heijin, who is now finishing medical school across the street, and she is very missed from the lab. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Heijin Kim.
Congratulations. Good luck for the future. Well done.
Kip Lacey received a Bachelor and a Master's of Science from the University of Georgia, where he worked on the population genetics of fire ants, which are renowned among locals for ruining picnic parties with their burning stings. He joined the Rockefeller graduate program in 2018. In my lab, Kip studied how clonal raider ants manage to reproduce asexually and effectively clone themselves. Asexual lineages arise relatively frequently across the tree of life via modifications to the meiotic program. However, we rarely understand how this actually works and what the genomic consequences of asexual reproduction are. Kip discovered that clonal raider ants employ a systematic violation of Mendel's law of random chromosome segregation during meiosis to produce unfertilized diploid eggs. In particular, these ants faithfully inherit pairs of chromosomes that underwent crossing over in recombination during meiosis 1. This surprising cellular memory of crossover status allows them to propagate without a notable deterioration of the genomic content. Kip's discovery is particularly exciting because we have reason to believe that similar mechanisms are also at work in many sexually reproducing organisms, although they would be much harder to detect there. Kip also found that clonal raider ants have in fact retained the capacity to reproduce sexually, at least in principle. Even though sexual reproduction seems to be exceedingly rare, it might help the species escape genome deterioration even over evolutionary timescales. It is hard to imagine the lab without Kip. His desk is right next to my office, and I can always count on being entertained by one of his witticisms or a stimulating conversation about genetics when I need a distraction. Kip will join the lab of Andres Pendeski, an alumnus of our graduate program at Columbia University, as a postdoc to continue his studies of exotic facets of inheritance, in this case, the phenomenon of genomic imprinting in mice. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Kip Lacey.
Thanks, Daniel.
Well done. Wonderful.
Gives me great pleasure to introduce Tyler Louie, an outstanding young scientist. Tyler did his undergraduate training at the University of California at Davis, where he worked on human immunodeficiency virus. He then moved to Hamilton, Montana, and spent 2 years at the Rocky Mountain Laboratory studying tick-borne flaviviruses with Marshall Bloom. Tyler arrived at Rockefeller with his then girlfriend, now wife, Becca, and their Australian shepherd, Scout. He had his own plans for his thesis work, the study of viral encephalitis, initially focusing on West Nile virus, which is a mosquito-transmitted RNA virus, which arrived in the United States in 1999 and is now an endemic cause of human cases of encephalitis. The only problem was that we weren't currently studying viral encephalitis. Undaunted, Tyler wrote and secured an NIH grant to pursue his ideas. He also set his sights on learning more about the central nervous system, given the rich neuroscience environment here at Rockefeller. Early on, Tyler noticed that the sites of virus infection in the brain differed depending upon whether or not the virus was delivered by direct infection of the brain versus infection in the periphery and subsequent neuroinvasion. This raised the question of whether early virus infection-induced signals in the periphery might be able to somehow warn the brain that an infection was underway and promote an antiviral response that could protect the central nervous system from lethal encephalitis. In an elegant series of experiments, Tyler went on to show that a protective response was in fact mediated in part by the antiviral cytokine interferon, and the cells at the blood-brain barrier were important for transmitting this protective response. Tyler's work raises many questions for future exploration about how signals in the periphery alert responses in the brain to protect this essential organ. Besides science, Tyler loves the mountains and will be returning to Rocky Mountain Laboratory for postdoctoral training and probably some hiking as well. This was all part of the plan when he arrived at Rockefeller and capped off by the recent arrival of his son, Emmett. Tyler's unquenchable curiosity and fearless exploration of the unknown will be greatly missed by me and the entire lab, but we look forward to following his next round of scientific accomplishments and especially visiting him in Montana. Mr. President, Mr. Vice Chairman, and honored guests, it's my pleasure to present to you Tyler Louie.
Congratulations, Tyler. Well done. Good luck. Thank you.
Good afternoon. Rico Lin grew up in Beijing, China, and came to Rockefeller as a graduate student after completing undergraduate degree at Cornell University. For her Ph.D. project, Rico made a big pivot from her undergraduate work studying host pathogen interactions in plants and shifted her attention to mammalian adipose tissue development and remodeling. A major focus of my lab is the biology of Beija adipocytes, a highly plastic cell type that in response to cold exposure can induce a thermogenic program, allowing these cells to convert chemical energy into heat. While this has become a field of research in its own right, Rico realized that many fundamental questions remained unanswered. Like, where do beige fat cells come from? Where do they go over the lifespan of an animal, and what exactly do they do? Over the course of her PhD work, Rico made two major advances that address these questions. First, she showed that beige adipocytes arise from a dedicated pool of progenitor cells during a key time window in early development. These cells arise in a programmed manner within the tissue niche, and their development is dependent on a postnatal surge in thyroid hormone. These cells then become thermogenically inactive or dormant as animals mature, but are reactivated in adult animals upon cold exposure. Second, Rico showed that these dormant beige adipocytes, while morphologically indistinguishable from white fat cells, have a unique molecular identity and play a key role in suppressing adipose tissue inflammation and in maintaining proper tissue homeostasis. I'm particularly proud that both of Rico's discoveries started with observations that did not match the dogma in the field. Rather than being daunted by this, Rico used these puzzling findings as a starting point and then meticulously worked out the underlying mechanisms. After graduation, Rico plans to pivot again for postdoctoral training at the interface of metabolism, neuroscience, and evolution. We will all miss Rico's fearless and inspiring approach to science, her leadership in the lab, and her eclectic set of hobbies that were memorialized on a graduation cap made by members of my lab that you will not see today. Rico has a very bright future ahead. Mr. President, Mr. Vice Chairman, and honored guests, it's my pleasure to present to you Dr. Rico Lin. Congratulations.
Lindsay Lopes received a Bachelor of Science from Haverford College in Pennsylvania and conducted research on sleep in the worm C. elegans at the University of Pennsylvania before joining our graduate program in 2018 and my laboratory in 2019. Initially, Lindsay set out on an ambitious project to develop neurogenetic tools that would allow her to identify and trace some of the neural circuits that underlie social behavior in ants. Unfortunately, about a year into her tenure in my lab, that project became a victim of the COVID pandemic and associated restrictions on laboratory work. One day in 2020, Lindsay and I sat outside on campus socially distancing and discussing how to proceed. In particular, we had gotten interested in the possibility that instead of focusing on a particular circuit, we could simply attempt to reconstruct the entire ant brain at synaptic resolution using electron microscopy, something that had recently been achieved in the fruit fly. I think we were both a bit naive about the scope of such an undertaking, which was good because we decided to jump right into it. Of course, the project required several external and internal collaborators, but Lindsay took the lead from day one, and thanks to her, you can now click through a digital representation of the ant brain and reconstruct any neural circuit you want. This project has already spawned many new collaborations for Lindsay, and it has created a lot of excitement in the neuroscience community. Through her tenacity, rigor, and resourcefulness, as well as her great social skills and community building, Lindsay has achieved a truly monumental feat. In addition to her scientific achievements, Lindsay has been a force for good, both as a co-chair of the Rockefeller Inclusive Science Initiative and as a mentor and colleague in my lab. Her social awareness and her drive to improve conditions for everyone have been invaluable. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Lindsay Lopes.
Congratulations, everybody. Well done. Let's take a picture.
Thank you so much.
Good luck.
Thank you.
I'm so very excited to be here today to introduce Elif Magemizela Brewer. I first met Elif several years ago when she joined our lab as a summer student. Working together with a postdoc, Sean Wallace, she sought to understand aspects of innate immunity in our favorite model system, the nematode C. elegans. Her clarity of thought and scientific curiosity were on full display already then, and when she decided to attend Rockefeller and to join my lab, I knew we had recruited an amazing scientist. As a graduate student, Elif initially pursued studies of memory formation in C. elegans, spending days and nights trying to understand how these critters remember the temperature of their incubation. However, as often happens in scientific pursuits, the project hit a technical snag, but in characteristic aplomb, Elif brushed aside the roadblock and set her eyes on a different goal, which was to constitute her thesis work. In our brain, half the cells are neurons, through which electrical signals zip along at breathtaking pace, allowing us to sense the world around us and to respond meaningfully and appropriately, and sometimes not so appropriately. However, these cells are only half. There are another 100 billion cells or so called glia or glial cells that physically touch neurons, but which remain in many ways mysterious. What do they do? What are they there for? This is the problem that Elif tackled, again turning to the worm, who, unlike us, has only 56 glial cells, giving us a fighting chance to understand something about what they do. Elif dove into her new project, generating information at breakneck speed. She honed her efforts on a particular glial cell in the head of C. elegans, showed that it can affect physiology of neurons with which it interacts, and uncovered a new set of molecular players, proteins, that engage in intricate communication between the glial cell and its associated neurons. The sheer amount of information she generated set a record for the longest thesis my lab has ever produced. In the end, Elif's work has, I believe, fundamental implications for what it is that glial cells do. Her studies revealed a question that no one even knew to ask, and her demonstration that what had been previously viewed as passive and generic properties of neurons were actually regulatable and by glial cells has far-reaching consequences. And although there remains much to learn about how and why all of this happens, Elif's work is an important, crucial step. As a lab head, my favorite moments are the intellectual tete-a-tetes with the researchers in my lab to understand the experimental results they have generated, to make models, to speculate, and to think big. And Elif, as the consummate scientist that she is, was a worthy conversationalist, always happy to engage, never shying away from telling me I was wrong, always politely. Those moments are what makes science special, and Elif provided them in spades. We are lucky that Elif will stay with us over the next year to complete her exciting studies, and I know that whatever she chooses to pursue after this time in my life, in my lab, will be extraordinary. It's a big time in my life, too. Tebriklar, Elif. Mr. President, Mr. Vice Chairman, and honored guest, it is my pleasure to present to you Elif Magemizolu Brewer. Congratulations.
Great job, everyone. You're more than halfway there. Keep it up. Wanbin Mei hails from China, where he studied mathematics and biology at Peking University. He came to Rockefeller intent on applying computational methods to the study of cancer. In my lab, Wanbin has been trying to understand why breast cancer spreads, or in scientific terms, why it metastasizes. For decades, scientists believed that this deadly process was driven by random mutations that occur in tumors. And for decades, scientists have searched tirelessly and spent hundreds of millions of dollars trying to find those specific mutations that might explain how cancer spreads. But despite all that effort, we've come up empty-handed. Wanbin had a bold idea. What if the ability of breast cancer to spread isn't just about random mutations in the tumor, but it's something inherited, something passed down in our genes? So he started digging into many genetic databases of breast cancer patients, and remarkably, he found something no one else had seen before. A common genetic alteration, something people are born with, that is linked to worse survival in 4 different breast cancer patient cohorts. He tested this gene in mouse models and discovered that it causes cancer to spread more aggressively. He then built a special mice that carried this human genetic alteration. And just like in people, those mice had more metastatic breast cancer than mice that lacked this genetic alteration. The genetic alteration he had identified was an amino acid change in a protein called PCSK9, which some of you may have heard about in the context of cholesterol. Wanbin found that this alteration activates a previously uncharacterized program independent of cholesterol in breast cancer cells that makes the cells divide rapidly in organs that the cells have spread to. Wanbin showed that if you block this protein using a drug, one that's already approved for cholesterol reduction, he could reduce the spread of breast cancer in mice. Finally, through a collaboration with Scandinavian scientists who had genetic data on a large collection of women with early-stage breast cancer, he found that women who inherited this variant had higher risk of their cancer spreading, breast cancer spreading. Wanbin's work reveals that our inherited genetics, which exist before the tumor forms, can impact the trajectory and outcome of a future cancer. Wanbin is headed to Boston for postdoctoral studies in cancer genetics at the Dana-Farber Cancer Institute. Wanbin, we will miss your brilliance, resourcefulness, and unmatched sense of humor. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Wanbin Mei.
Congratulations. Good luck. Thank you. Thank you. Thank you. Thank you.
Hello again. Oops. Dennis Melendez received a bachelor in psychology from Hunter College here in Manhattan, where he worked on vocal plasticity in songbirds with Ofer Chernikovsky. He then came to Rockefeller for his graduate work in 2017.
Being a true ethologist at heart, Dennis joined my lab to explore the collective behavior of the clonal raider ant, my lab's main study system. Through meticulous observations, he discovered that colonies of the species are nomadic. In other words, they frequently relocate to new nest sites, a behavior that had not been observed before. How groups of organisms decide when and where to move without losing contact is an important question in collective decision-making, and Dennis's discovery opened the door to detailed laboratory studies of this problem. Nest relocation and the underlying collective processes had been previously examined in honeybees and acorn ants. However, these species hunt for new homes only sporadically. In clonal raider ants, nest relocations turned out to be the norm, with true nomadism being an integral part of the species' life history. Furthermore, Dennis was able to show that clonal raider ants employ very different mechanisms during collective decision-making compared to other species, demonstrating how evolution works within the framework of a given species' biology to produce convergent solutions to shared challenges. During his time at Rockefeller, Dennis has been prolific and active outside of the lab as well. Not only did he and his wife have two extremely cute kids, Dennis also emulated the subjects of his studies by repeatedly buying and selling houses and moving all across town. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Dennis Melendez.
So Chad Morton grew up in St. Kitts, a beautiful little jewel of an island in the Caribbean. The path that brought him to our little island, the island of Manhattan, was not an easy or conventional path, but Chad is not a conventional student. From the first day we met, I was immediately impressed by his tireless energy, ambition, and determination to seize every opportunity. From leaving St. Kitts to pursue science, which in itself was a daring departure from tradition, to studying chemistry at Southern Connecticut State University, attending a pre-baccalaureate program at Northwestern before eventually enrolling as a graduate student here at Rockefeller. Quite fittingly, given Chad's circuitous path into science, his thesis explored the neural and behavioral algorithms of navigation, the strategy animals use to travel from one distant location to another. So for many species, odors serve as critical navigational cues, chemical beacons that allow them to travel long distances towards a remote source. However, because odors are invisible, this process has been really challenging to study. Chad overcame this technical hurdle in a spectacular manner by developing a virtual reality paradigm that allowed him to record both neural activity and behavior as tethered Drosophila fruit flies navigate an odor plume, revealing an unexpected strategy in which flies weave into and out of the plume, updating a short-term memory of how to return back to the odor plume whenever they exited it. Chad's work showed that Drosophila use odors as dynamic landmarks, suggesting a surprisingly sophisticated memory-based computation for navigation and opening up new inroads to defining its underlying neural basis. But beyond Chad's scientific creativity, what I really admire most about him is how generously he shares his passion for scientific discovery with those around him, always eager to bring others forward with him along his journey. Chad co-directed RISI, the Rockefeller Inclusive Science Initiative, during a particularly challenging period that ultimately helped to galvanize several meaningful changes on campus. And during that period, Chad emerged as a leader, a clear leader in developing community initiatives, and was very appropriately honored with the inaugural Inclusive Excellence Graduate Student Award last spring. So Chad now serves as a program officer here on campus in Rockefeller's Office of University Life and Community Engagement, where he continues to be a vibrant presence on campus. And I'm so grateful that I have the chance to continue to meet with him. But I suspect Chad's most meaningful work is yet to come as he sees his dreams of capacity building in the Caribbean come to life. Last year, Chad and his wife Hillary launched a program called Inspired that offers high school students the opportunity for scientific discovery on his own beautiful little island of St. Kitts, using DNA barcoding to begin cataloging the island's amazing biological diversity. Ultimately, his aspiration is to make scientific island hopping much easier and more direct for future generations. So I think as every graduate student and even faculty member here today can appreciate, it is very easy to get mired in the inherent frustrations and challenges that come from truly making insights into biological mechanisms and get stuck in fretting over the uncertainties of the future. But what Chad continually reminded me, and continues still, is that being a scientist is not a challenge, but really a privilege to explore and illuminate the deepest mysteries of biology. And this is something we should never forget, especially at this moment in history. Pursuing science at Rockefeller as we do is indeed a privilege, but is never more rewarding than when you can share it with students like Chad, who bring such generosity, passion, perspective to every conversation. So, Mr. President, Mr. Vice Chairman, and honored guests, it is my great privilege and pleasure to present to you Chad Morton.
Congratulations, Chad. Good luck.
So when Sandra Nakandakari-Higa joined our lab, we had just published a new technology that we had created for labeling cell interactions in live mice that we called lipstick. And although this worked very well, it had a key limitation. It only worked for the interaction, particular interaction between antigen-presenting cells and helper T cells. Sandra's ambitious task was to make this system useful to everyone, so lipstick for the masses. With a tour de force effort, she developed what we now call the universal lipstick system, which works as an in vivo recorder for interactions between multiple types of cells, both immune and non-immune in origin, and across many organs of the mouse. And the community is highly appreciative of her effort. There's no place I go now anywhere in the world where there isn't someone using U-lipstick for one thing or another, which is a real testament to Sandra's impact on the field. And this was only half of her PhD thesis. But as impressed as I am by her scientific feats, I'm also impressed by her athletic feats, which include winning both the Rockefeller spikeball and pickleball tournaments, running a half marathon in 1 hour and 46 minutes, going back to her native Peru to compete in the 2025 competition for Latin American Japanese athletes, and biking to her lab retreat in Cape May, which is 160 miles away, and on her way winning money in Atlantic City. And all of which was fueled by about 10 cans of Pepsi a day and not of the diet type. So Sandra, I wish you the best of luck in your future career and keep running both physically and scientifically. So Mr. President, Mr. Vice Chairman, and honored guests, it's my pleasure to present to you Sandra Nakandakari-Higa.
When you grab a strap on a train or subway or bus, and I hope you have such experience, you hold on it tightly only when you sense a force. A similar mechanism engages when chromosomes are distributed to dividing cells in mitosis. Each chromosome tightly grabs the polymers called microtubules, but if chromosomes were pulled to the wrong direction, the attachment must be released. How can chromosome distinguish between correct and wrong attachment has been a big mystery. This was the question Emin Nu tackled. Soon after Emin moved from China to Cornell University, he became addicted to structural biology. Emin found that in structural biology, there are clear answers to solve, and he can keep pushing the resolution, which means obtain the better answers. It has been known that the kinetochore protein complex NDC80 grabs the microtubules, and the phosphorylation of the NDC80 releases from microtubules. By developing a new cryo-electron microscopy analysis method, Emin found that the structure of MDC80 is rearranged upon binding to microtubules such that it became resistant to phosphorylation. Emin's discovery forms a structural basis on how chromosomes can sense the microtubule attachment. The path Emin took was not a smooth one, however. He has been separated from his loved ones in his home country since the pandemic. He never returned home since then. He had to completely change his research field during a Ph.D. program. And his mentor kept asking demanding tasks because he was very good at solving difficult questions. And he was lucky to me staying in my lab for another year. Despite these difficulties, Emin's passion to science has never been eroded. As a talented and creative structural biologist, Emin has been generously offering his helps to others, and so visitors constantly came to the lab to talk to him. I'm confident that Emin will keep helping our society by solving difficult questions as well as by serving elaborate Chinese meals when he has access to the nice kitchen. Mr. President, Mr. Vice President, and honored guest, it's my pleasure to introduce to you Emin Nu.
It's my pleasure to present Jesse Novak, reading the tribute from his thesis advisor, Elaine Fuchs. I've had the pleasure to mentor truly special young scientists through their graduate education. In many different ways, they've shined as rising stars. Jesse is unique in that he's managed to ski the Matterhorn, travel around the world, stay fit with weightlifting, and carry out beautiful Ph.D. research in my laboratory. Oh, and he happens to also be fluent in Spanish and Chinese in addition to English. Jesse received his BA in molecular biology and Chinese from Middlebury College, Vermont, where he studied synaptic transmission in C. elegans in the lab of Dr. Glenn Ernstrom. He spent his summers in New York City with Nodfeld and then Signoretti labs, and became increasingly more interested in biomedical research. In my lab, Jesse became fascinated with the metabolic control of cancer. Another MD PhD student in the lab had just discovered that when mice bearing non-melanoma skin cancers are placed on a serine-deficient diet, their tumors shrink. Jesse wondered what happens to normal skin in the face of serine deficiency. Interestingly, the stem cells of the hair follicle respond to malnutrition by activating a pathway known as the integrated stress response. Jesse further discovered that when the skin is wounded, the stress pathway is also elevated, and when skin is wounded in mice experiencing malnutrition, the pathway soars. Jesse found that remarkably, the tissue stem cells measure stress levels, and they link them to deciding what they should do and when. Through his studies, Jesse uncovered an amazing evolutionarily conserved program that probably all tissues use in times of stress to funnel their tissue-making skills into those tasks which are most vital to life. His work is now in press in cell metabolism and is already garnering major attention in the field. It has been a pleasure mentoring Jesse through his PhD studies. He has held true to his passions and goals throughout the time I've known him. He excels at everything he does, and his science has been as dazzling as his worldly adventures. Mr. President, Mr. Vice Chairman, and honors guests, it's my pleasure to present to you Jesse Novak. Congratulations.
Thank you.
Thanks for all you do.
Thank you.
Cells in the body physically interface with their local tissue environments. These mechanical interactions are mediated by the actin cytoskeleton, a network of proteins which assemble into cell-spanning scaffolds that generate and detect forces. Like all construction materials, the cytoskeleton is subject to wear and tear, and physical damage of cytoskeletal networks can lead to their collapse, compromising their functions. Unsurprisingly, gene networks have evolved which detect and repair cytoskeletal damage, but it has been unclear how the proteins they encode work together to find and fix mechanical defects at the molecular scale. Donovan Foy's thesis research markedly advanced our understanding of the system. In a biochemical and biophysical tour de force, Donovan reconstituted the repair reaction from 6 purified proteins, including force-generating myosin motors. He found that the key repair protein zixin forms force-dependent polymers, which can temporarily bridge ruptures in the rope-like actin filaments which hold the cytoskeleton together. These zixin polymers bind additional proteins which trigger the local assembly of new actin filaments, as well as factors which stitch the filaments together into multi-stranded cables, reinforcing the network at these mechanical weak points. His work highlights how cytoskeletal damage repair occurs at the level of many protein networks, rather than binary interactions between individual factors. This provides a satisfying explanation for how nanoscale protein molecules can coordinate cellular mechanical homeostasis at length scales 1,000-fold greater than their own size. Beyond understanding how the physical integrity of our cells is maintained, Donovan's work may also have implications for the design of self-healing biomimetic materials. Donovan is a true force of nature at the bench, and he has also pushed forward many other projects which will keep my lab busy for years to come. He has also been very active in promoting social justice in science as a member of RISI, while also seeking to broaden participation in science through RockEDU outreach programs. He still somehow finds time to maintain an intensive schedule of outdoor rock climbing excursions, as well as his most recent adventure as a dog parent to the adorable Italian greyhound Milo, alongside his partner Kenny. Many exciting new discoveries are sure to come as Donovan is next pursuing a postdoc studying the role of forces in plant regeneration at Caltech. Mr. President, Mr. Vice Chairman, and honored guests, it is my great pleasure to present to you Donovan Foy.
Congratulations. Very good. Good luck. Thank you. Thanks, Greg.
Patrick Piekarski received a Bachelor of Science from the University of Calgary and a Master of Science from the University of Central Florida, where he did beautiful work on the molecular phylogenetics and the evolution of female casts and wasps. He joined our graduate program in my lab in 2018. At Rockefeller, Patrick has studied the question of how genotypic, social, and environmental factors affect the development of individuals into different castes. In ants and other social insects, female larvae can usually develop into at least 2 castes, the workers, which are small, short-lived, don't lay eggs and maintain and expand the colony, as well as the queens, which are large, can live for many years, and are specialized egg layers. This opens many interesting questions in evolutionary and developmental biology, as well as epigenetics. Patrick's thesis makes several major contributions to these important questions and tells a beautifully comprehensive story encompassing careful experiments and analyses of morphometric data all the way down to the genetic details of how caste traits are determined at the molecular level. For example, Patrick has achieved the first global description of how the genotype of caregivers affects the development and well-being of their offspring. Patrick's patience, experimental rigor, statistical prowess, and excellent writing skills have made it an absolute pleasure to work with him, and I admire him particularly for his ability to deal with frustrating reviewer critiques. He takes the good and rebuts the rest with calm authority. While others go and scream into the void or pillow, Patrick has a similarly noisy but more productive outlet. He's a big metalhead. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Patrick Piekarski.
Thanks.
Amanda Shilton. It is my great pleasure to introduce to you Amanda Shilton. Amanda was born and raised in Zimbabwe. Amanda does not come from a privileged background, and her parents had to work really hard to give her the opportunity to study abroad. She took this opportunity and moved to Boston to attend Brandeis University. She graduated summa cum laude in 2018, completed a master in neuroscience, and was accepted to the Rockefeller Ph.D. program. She joined my lab in 2019. So for the third and last time, bacteria are infected by viruses, and one of the immune systems that they have to defend themselves is called CRISPR, and that's what we study in my lab. There are many types of CRISPR immune systems, and Amanda decided to study the arguably most complex of these systems, known as type 3. She used not only genetics and next-generation sequencing, both approaches that we commonly use in the lab, but fluorescence microscopy and cell sorting, new techniques that she brought to the study of CRISPR, to show that type 3 CRISPR systems provide immunity by attacking the host to prevent the spread of infection instead of attacking the virus directly, as the other CRISPR systems do. Her work was best described by a reviewer of her paper, whom stated that the work provides an unprecedented level of detail on the function of type 3 CRISPR systems in vivo. Amanda did not forget her struggles to become a scientist and is very committed to help others. In the lab, she has always been the champion of inclusivity, and she has contributed greatly to create a very positive environment. I wish her the best as she continues her postdoctoral studies at Duke University to investigate the maternal-fetal interface and how the placenta prevents infections. Mr. President, Mr. Vice Chairman, and honors guest, it is my pleasure to present to you Amanda Shilton.
Congratulations. Well done. Good luck, Amanda.
It's my pleasure to present Alexander Stewart, reading the tribute from his thesis advisor, teacher de Lange. Alex defended his thesis a year ago, but is still in our lab as a postdoctoral fellow to do the experiments necessary to satisfy the reviewers of his paper. For his thesis project, Alex had decided to revisit several fundamental questions about replicative senescence. The mortality of our cells, their inability to divide beyond a certain set point, is due to telomere shortening and and this process of replicative senescence represents a powerful mechanism by which our bodies block the outgrowth of early cancer clones. Most people in the field thought that cellular senescence was fully understood and that no questions remained. Nevertheless, Alex decided to dig into some of these supposedly solved questions and not only change the way we think about how our cells senesce, but also showed why our cells have a much longer replicative lifespan under low oxygen conditions. Alex showed me and others in the field that if you look carefully and closely, you can discover new things in an area that was supposedly thoroughly picked over. Unlike the primary cell cultures that Alex miraculously managed to keep alive and uncontaminated for many months, if not years, it takes for them to undergo senescence, Alex did not slow down during his thesis work. He did not show hallmarks of aging or diminished ability to produce or reduce fitness. Rather, Alex became better, more productive, more insightful, and more fit for purpose, not only at the gym, but also at the bench and on stage at various meetings. I can confidently say that I've never seen a student grow to scientific prowess the way I saw Alex blossom. Once Alex's paper is accepted, he will likely move to Boston to a postdoc at the august institution that is fighting for its life and for the life of scientific endeavor in general at the moment. We'll miss him terribly, but his future lab will gain a smart, inspiring, funny, and warm colleague. Mr. President, Mr. Vice Chairman, honored guests, it's my pleasure to present to you Alex Stewart. Congratulations.
Thank you.
Andrea Terceros, Andrea grew up in Bolivia, moved to Canada, and had just graduated from McGill University when I first met her as an incoming Ph.D. student here at Rockefeller. She came to do a rotation with us, but the chances were pretty low that she would join my lab. She wanted to study simple and tractable model systems like fruit flies, and she wanted to work with the greats, like Mike Young and Leslie Vossel. And here I was, a newly minted faculty member, working in a not-so-simple or tractable model system, the mouse. Naturally, I worked really hard to recruit Andrea. I told her, these other labs aren't so great. We're obviously doing much more interesting things. Somehow, I got her to join my lab, and I consider this one of my biggest accomplishments, because over these past 6 years, I've easily learned more from her than she's learned from me. She has a calm and patient, yet deeply creative and rigorous manner about her that has been an enormous inspiration to me. For her thesis, Andrea focused on addressing a deep and challenging problem. Why do some memories persist while others are forgotten? Andrea brought an elegant solution to break open this problem by first developing new ways to study mouse behavior. Using automated and scalable platforms where she could continuously monitor which memories are being formed, maintained, and forgotten over time. She then applied new methods that allowed her to track what molecules are changing in the brain, and in particular to focus on the ones that correlate most with memory persistence. These were extremely challenging experiments, and most impressive to me was not only Andrea's boldness to pursue these efforts, but her nimbleness to pivot when needed, to let go of directions that seem futile, and instead embark on alternative strategies, one of the hardest things to do as a scientist. She repeatedly abandoned techniques that lacked sufficient resolution, or she was not happy with the quality of the data, and she reinvented the question so she could start anew. Eventually, she and her collaborators landed on what I think are a remarkable series of discoveries, that the brain assigns value to memories as they're being formed, turning on a sequential set of molecular timers that allow memories to be maintained for longer and longer periods of time. These molecular timers, it turns out, are some of the same molecules that are being used during developmental memory, as organs are being formed and maintained, and during immune memory, when priming with an initial infection speeds up the response to a subsequent infection. Thus, she provides a view where the very same molecules that are being used to maintain cellular memory elsewhere in the body are being co-opted by the brain to support long-lasting cognitive memories. And perhaps most strikingly to me, she demonstrates that memory storage is not a one-time process, but rather an active, sequential, and continuous process that is required to keep committing memories into progressively more enduring forms. I'm so proud of her scientific journey and her remarkable findings. We'll miss her so much, her warmth, her curiosity, and her scientific drive. Mr. President, Mr. Chairman, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Andrea Terceros.
Congratulations. Well done. We're very proud of you.
Good afternoon. When our proteins don't work properly, or if they form aggregates in our cells, they need to be selectively cleared. This process is essential for life and depends on VCP, a powerful molecular machine that processes these proteins for recycling or their destruction. Consistent with this important function, mutations in VCP that alter its activity have been shown to cause diseases such as Alzheimer's. Lauren Vostel, an outstanding student in the tri-institutional Ph.D. program in chemical biology, helped design drug-like chemicals that can selectively stimulate VCP's activity, potentially correcting its cellular function. In parallel, to examine how VCP's function is regulated, Lauren developed and applied a powerful photochemistry-based approach to profile proteins that bind VCP in living human cells. Using cryo-electron microscopy, Lauren determined in atomistic detail how VCP's activity may be coordinated with a protease, a VCP-binding protein she identified. Finally, she designed biochemical assays to decipher how VCP and this protease function together and thereby ensure the clearance of damaged proteins and pathogenic aggregates. After completing her thesis research, which was in part supported by the Kellen Entrepreneurship Award, Lauren has joined a major pharmaceutical company where she is applying her skills to develop new therapeutic approaches. While she worked with us, we greatly admired Lauren's creativity, intellect, tenacity, and discipline. We will miss her infectious enthusiasm for innovative chemical biology and look forward to celebrating her completing many more New York City marathons. We may not miss her uncanny ability to repeatedly crush us, and I'm told even ChatGPT, in our lab's annual March Madness pool. We wish her all the best and look forward to her continued success. Mr. President, Mr. Vice Chairman, and our honored guests, it is my distinct privilege to present to you Lauren Vostel.
Sure. In the field of chromatin research, there are two ways of looking at the genome. One is the three-dimensional view in which we see DNA wrapping around bundles of histone proteins called nucleosomes and being organized into larger domains, groups of nucleosomes, or even different categories like heterochromatin and euchromatin. The other is the sequence view, in which we map the locations of proteins, gene activity, and other processes onto the linear DNA sequence of the chromosome. 3C, or chromatin conformation capture methods, attempt to link these two ways of looking at DNA. But unfortunately, the data they produce is still very sparse compared to the vast scale of the genome and the vast complexity of chromatin. It is in some ways like the old story of blind men trying to understand an elephant by grasping different parts of it. How do you take these disparate and sparse observations and figure out how they fit into the bigger picture? Devaney West took on this formidable challenge. And an analogy that's helpful is to think about how do you understand an elephant using sparse data. It's a terrestrial tetrapod, so you might know that it has 2 ears and 4 legs and a tail and a head. So similarly, Devaney set out to incorporate information that we know about nucleosomes using Bayesian inference approaches so that she could use simulations that we can do in the lab and that other folks in the lab also contributed to, combining them with bioinformatics data from 3C approaches to be able to get much more interpretable and information-rich models of what chromatin actually looks like in different epigenetic states. So this has helped us think about how do we go from an abstract, sparse map of chromatin interactions to a real physical object that is chromatin, and it's helping us think about how DNA-based processes operate in the nucleus. Mr. Vice Chairman, Mr. President, and honored guests, it is my great pleasure to present to you Devaney West.
Congratulations. Well done.
Okay. Nori Yamaguchi hails from Japan, where he trained in medicine and oncology. His experiences in oncology motivated his pursuit of research into how pancreatic cancer cells spread and grow as metastases in the liver. It has long been known that pancreatic cancer cells can survive in extremely low oxygen environments, a trait that is essential for their ability to spread to the liver, which is connected to the pancreas through a low oxygen blood supply. Nori discovered that pancreatic cancer cells achieve this by activating a protein called neuronal pentraxin 1, or NPTX1. He found that cancer cells secrete this protein, which then binds to a receptor also identified by Nori on the surface of the same cells. This interaction triggers a signaling cascade that maintains a protein known as hypoxia-inducible factor 1, or HIF1, in the nucleus. There, HIF1 turns on genes that enable cancer cells to adapt to low oxygen. HIF1, a protein whose discovery was recognized by the Nobel Prize in 2019, plays a central role in cellular adaptation to hypoxia. And Nori's work has illuminated how pancreatic cancer cells keep it in the nucleus to preserve its activity during cancer metastasis. Building on these mechanistic insights, Nori developed an antibody blocking this interaction between neuropentraxin 1 and its receptor, and showed that this impairs the growth of pancreatic cancer cells under low oxygen conditions and reduces their ability to form metastases in the liver. Nori is now a clinical investigator and physician scientist at the National Cancer Institute, or NIH, where he continues to advance our understanding of pancreatic cancer cell metastasis. Nori, we deeply miss your intellect, boldness, and remarkable breadth of knowledge in both science and medicine. We're excited to follow your continued discoveries in this field. Mr. President, Mr. Vice Chairman, and honored guests, it is my pleasure to present to you Nori Yamaguchi.
This is the most critical part. If we get it wrong, you have to start over. Graduates, would you all please rise? By the authority of the University of the State of New York, the Regents of the University of the State of New York, which they have vested to the trustees of the Rockefeller University, and it is by them delegated to me, I'm delighted to confer upon you the degree of Doctor of Philosophy, which admits you to all the rights and privileges thereunto pertaining. Congratulations.
Well done.
Okay, please be seated. So now we will turn our attention to the 2025 recipients of the Doctor of Science honoris causa, Quarraisha Abdool Karim, Salim Abdool Karim, Marlene Hess, and Michael Young. These individuals have shaped the scientific and biomedical landscape in myriad ways through their vision, innovation, and leadership. It's a tremendous privilege to welcome and recognize them today. I'll begin by recognizing Quarraisha and Salim Abdool Karim. Following this tribute, we will hear in turn from Quarraisha and Slim. Growing up in apartheid South Africa, Salim, known as Slim, and Quarraisha Abdool Karim witnessed how societal inequalities sabotage health, education, and well-being. With unwavering persistence and courage, they have dedicated their careers to improving the lives of vulnerable people through targeted research, evidence-based policy proposals, public education, and wise activism. The Abdool Karims have shown keen understanding of how to apply their talents most effectively and an internal compass that has unerringly pointed toward truth and justice. One can often learn much from about an adult from traits they expressed in childhood. For Slim, it started with his childhood dresser. Socks needed to go where socks belonged. Shirts needed to go where shirts belong. He didn't want to waste much time looking for things. Everything had its place. His desk told the same story, with books and papers organized for ready access. Unfortunately, his mother occasionally made the mistake of dusting his desk, and when she did, she removed all of the carefully systematized piles of books and papers that ended up almost randomly distributed on his desk. I understand Slim was never pleased with this disturbance. But his strong inclination toward order reflects an exquisitely organized mind that is clearly seen in his adult life and has directly contributed to his success in politics, medicine, and communication. Slim is well known for his uncanny ability to hear a rambling discussion and then crisply summarize it, articulate the salient points, identify the relationships between seemingly discrepant thoughts, and suggest a course of action. His knack for identifying and breaking impasses in negotiations has made him a brilliant leader. Perhaps unsurprisingly, engineering appealed to Slim, but he could not afford the university's registration fees. Undeterred, he showed up for classes anyway. But on day 3 of classes, he came home to find an envelope from the University of Natal Medical School, which offered him a full scholarship to study medicine. Ever practical, he immediately pivoted to this new career path. Early in his professional training, he began harnessing his skills and sensibilities. As a third-year medical student, he published his first paper about how apartheid was creating differential health status across race and geography. He subsequently co-created an anti-apartheid doctors organization. After finishing his degree in 1983, he did a year of research on hepatitis B virus and decided to continue in the field of virology. I will return to Slim. But if a bedroom dresser illuminates a deeply ingrained feature of Slim's personality, an electrical outlet may reveal one of Quarraisha's. When she was very young, she discovered electricity by sticking her finger into a socket. She was so excited about the sensation, she encouraged her little sister to try it too. Quarraisha's parents quickly realized that she needed more stimulus to satisfy her insatiable curiosity, so they sent her to school at age 3. Quarraisha came from an Indian community whose collective livelihood depended on the labor-intensive agricultural economy. People sacrificed from their small salaries to support schools for the next generation, as they believed that education would be the great equalizer. Quarraisha benefited from that practice. Beyond knowledge acquisition, she absorbed the importance of lifting the community. Her teachers recognized that she picked up knowledge very quickly and had a flair for sharing it. They routinely asked her to teach students who were lagging so the entire class could move ahead. In 1976, Quarraisha's final year of high school, her family bought a TV, and the visuals that it offered provided new perspective for her about South African life. She saw the Soweto uprising, school children protesting instruction in Afrikaans, a derivative of the adult language and associated with apartheid, and a foreign language to most that created a barrier to learning for Native Africans. The police responded to these protests with brutality. During college at the only university for Indian students in South Africa, Quarraisha juggled protests with academic studies. By the time she graduated, she had developed an interest in immunology, so she embarked on research in that area. Other universities began to open up racially, and she decided to earn a second bachelor's degree. Growing increasingly concerned about how poverty contributes to poor health, she began working on weekends at an all-volunteer mobile rural community health center that provided free services. In parallel, she continued her experimental work on blood disorders. In 1987, Quarraisha, by then a research scientist, met Slim, then a medical resident in clinical virology, when she needed to use an ultracentrifuge that was only available in Slim's department. He then appointed himself as custodian of the machine to ensure that their paths would frequently cross. In the meantime, Slim had received an invitation to do a fellowship in epidemiology at Columbia University. Mervyn Susser, a South African expatriate who, with his wife Zena Stein, led that department and wanted to train black epidemiologists. He had contacted one of Slim's professors for recommendations of up-and-coming candidates, and he suggested Slim, and Slim immediately accepted the offer. When he left for the U.S., he and Quarraisha kept in touch by writing letters. Soon, poetry crept into these exchanges, and within months, they married, and Quarraisha moved and joined Slim in Manhattan. Under Stein's mentorship, she dove into master's degree work in public health at Columbia as the AIDS epidemic was raging in New York City. The Abdool Karims watched the plague convulse the city, and they absorbed information about it in seminars. When AZT was approved by the FDA in 1987 as the first antiretroviral drug, patients with HIV/AIDS suddenly began living longer. In 1988, they returned to South Africa, determined to study HIV there, which had as yet produced few cases of AIDS. But their experience in New York suggested that the virus was likely spreading unnoticed and that AIDS would soon begin emerging. They decided to do a community-based survey to find out the prevalence and demographics of HIV infection there. To their surprise, in contrast to the US, where 90% of HIV infections occurred in males, they found that 75% of HIV infections in South Africa were in young women. Furthermore, the prevalence peaked in the teen years in women, whereas it didn't show up in men until nearly 10 years later, leading them to infer that older men were having sex with and infecting much younger women. The Abdool Karims wanted to develop ways for girls and women to avoid HIV infection that did not rely on male cooperation, as condom use or mutual monogamy was often challenging for young women to negotiate with older men. For 18 years, they tested candidate agents in clinical trials involving thousands of participants, but each one of these failed. They then had a new idea. Most of the drugs that they'd assessed targeted HIV entry into cells. They then decided to evaluate a chemical that instead stopped the virus from replicating once inside cells. And in 2010, they completed a clinical study using a drug, tenofovir, first as a vaginal gel, then as an oral tablet that was stunningly successful in preventing HIV infection in women. This drug quickly became a central element of the World Health Organization's HIV prevention strategy implemented in many countries, including the US. Because the use of these episodic medicines proved challenging to use without fail, recent efforts have focused on development of long-acting drugs that could prevent HIV infection. And just last fall, they and colleagues demonstrated very high efficacy of the drug lenacapavir, which can be administered only twice per year as a subcutaneous injection and provides continuous protection. By the late 1980s, Africa bore the majority of the planet's HIV burden, and yet the International AIDS Conference had never been held there, so the devastation remained largely invisible to the world. Quarraisha spearheaded a quest to bring the 13th International AIDS Conference to Durban, South Africa in the year 2000. The rampant deaths in child-headed households galvanized the global community, which committed to providing antiretroviral drugs to Africa. But across Africa, most people with AIDS also had active tuberculosis. No one knew how to best treat individuals who were co-infected. Healthcare workers worried, for example, that co-administering drugs for AIDS and TB would have toxicity that would harm people. The Abdool Karims bravely organized and conducted a randomized controlled study to address this issue. They clearly showed that initiating combined TB and HIV therapies dramatically reduced the one-year mortality rate compared to delayed initiation of antiretroviral therapy. Within weeks of publication in 2009, the World Health Organization adopted new recommendations that reflected these findings. By 5 years later, annual deaths from AIDS in South Africa had plummeted. For decades, the Abdool Karims have continued illuminating HIV's heterosexual transmission cycle in Africa. Eventually, they sequenced thousands of viral variants to trace the virus's path through various populations. And as they had surmised, adolescent girls and young women contracted HIV from men who were on average almost 9 years older. Later, women who harbor the virus couple with male peers, and 39% of these men also have sex with much younger females. The Abdool Karims presented these results at the International AIDS Conference in mid-2016, and later that year, UNAIDS published a report that spotlighted the findings and the ramifications for countries in which men were driving the epidemic by infecting teenagers and young women. This document guided several African countries as they drafted new approaches by which to prevent and treat HIV. The Abdool Karim's conclusions had already propelled the United States President's Emergency Plan for AIDS Relief, PEPFAR, to direct resources toward adolescent girls and young women. The Abdool Karims also played a pivotal role in developing South Africa's scientific infrastructure. They launched the Center for the AIDS Program of Research in South Africa, or COPRISA, in 2002. COPRISA initially focused on HIV, but its scope has subsequently widened. When COVID-19 emerged, the Abdool Karims and their COPRISA colleagues focused on the sequencing capabilities that they had developed to analyze HIV and redirected these resources toward understanding SARS-CoV-2 variants that were the cause of COVID. In November 2021, they published the first description of a very unusual variant of SARS-CoV-2, now called Omicron, which was dramatically different from the initial variants identified and foretold a lethal wave of virus across the globe in the winter of 2022. Their paper garnered enormous attention, and its predictions helped shape the response to the ongoing pandemic. In addition to establishing COPRISA with Quarraisha, Slim led the founding of 4 other major research centers in South Africa and served as each one's first director to get it off the ground. To create these institutions, he won support from the Wellcome Trust, the NIH, the Howard Hughes Medical Institute, and the South African government. Through a Fogarty-funded collaboration with Columbia University, they have trained more than 600 South African HIV and TB investigators, and these scientists now underpin South African infectious disease research. The Abdool Karim's impact has spread as they have helped to build and connect HIV research centers in other nations, including Zambia, Nigeria, and Senegal. As if all of this were not more than sufficient, the Abdool Karim's influence has also extended into politics and policy. In 1999, Thabo Mbeki became the president of South Africa, succeeding Nelson Mandela. He disseminated false narratives about AIDS, contending that AIDS deaths arose from TB and malnutrition, not from the HIV virus. When his government then restricted the use of nevirapine, which reduces mother-to-child transmission of HIV, Quarraisha presented expert testimony challenging this restriction in the South African Constitutional Court. The court ruled the government was violating the rights of women and newborns and required the medication to be offered widely. On television, radio, and in print, Slim contradicted AIDS denialists, including the president. Later, they challenged COVID-19 anti-vaccination campaigns and debunked sham treatments such as hydroxychloroquine and ivermectin. He was a frequent communicator on CNN, The Voice of America, BBC, and Al Jazeera, and was known for delivering clear and accurate medical information during a rampant time of confusion, fear, and uncertainty, as he had also done during the AIDS crisis. The Abdool Karims have figured prominently in molding science policy across the planet. They have held senior advisory roles at many agencies and enterprises, including the United Nations. Excuse me. My apologies. They've held senior advisory roles at many agencies and enterprises, including UNAIDS, Africa's CDC, and PEPFAR. In 2022, the World Academy of Sciences elected Quarraisha as president. Leaders at the highest levels, including the UN Secretary General and the Director General of the World Health Organization, trust them to offer sage and honest counsel. Quarraisha Abdool Karim and Salim Abdool Karim, you are known and admired for your relentless dedication to preventing HIV infection and preventing death in those who become infected. You have saved countless lives worldwide. We applaud you not only for your scientific contributions, but also for your tireless humanitarian dedication. You've been at the forefront of addressing this public health crisis, devoting your careers to advancing science with a clear focus on global health equity. It is my great honor to confer upon you the degree of Doctor of Science honoris causa. Quarraisha, please rise first to receive your hood and diploma. And now, Slim, it's your turn. So, Quarraisha, would you please come and present your remarks?
Thank you, President Lifton, Vice Chair Steele, Dean Stearns, fellow honorary degree recipients Marlene Hess and Mike Young, faculty, students, family, and friends. Greetings and hearty congratulations to the graduating class of 2025. I'm deeply honored to be a member of the class of 2025. To receive an honorary doctorate from the Rockefeller University, an academic institution synonymous with a long history of leadership in excellence, innovation, and generating knowledge for the benefit of humanity, is a singular honor that I'm deeply grateful for and humbled by. This generous recognition is a powerful reminder of the transformative role of science in shaping a more inclusive and just world. It is truly a privilege to share this moment with each of you today, and it's also one that I share with my family and friends, teachers, mentors, research partners, collaborators, colleagues, and communities who have taught me, challenged me, and continue to walk alongside me. I accept this honorary doctorate from Rockefeller University as a recognition of our interconnectedness and shared humanity across the globe, recognition of the power of global solidarity and partnerships, and of excellence and innovation that is deeply embedded in our research on HIV prevention and treatment over the past 37 years. When the HIV epidemic first began sweeping through our communities in Southern Africa in the late 1980s, it quickly became clear that young women were at the center of this crisis. Figuring out why and how to respond to the needs of these young women became a lifelong commitment. In this journey, we've learned that science can transcend borders and divisions. For example, while Quarraisha provide the initial evidence that antiretrovirals prevent sexual transmission of HIV as pre-exposure prophylaxis or PrEP, Today, PrEP is used in over 100 countries. Further, if a patient with HIV and TB co-infection anywhere in the world, including here in New York City or anywhere in the USA, is being treated according to WHO or US guidelines, that patient's treatment is based on research conducted in South Africa by a multi-country research team. CAPRISA's contributions through science are small, but important steps in the larger social challenge of gender power dynamics, inequities and inequalities, but highlights the powerful role that science can play in social transformation. Through my mentors, I've also have come to appreciate more and more that real innovation also lies in how we invest in future generations of scientists and leaders. Our experience in training and mentorship of young scientists in Africa and in undertaking research responsive to the realities on the ground through CAPRISA and through the Columbia University Southern African Fogarty AIDS International Training Program has revealed the power and multiplier effect of nurturing of hundreds of scientists and leaders who are now shaping impactful research across the continent and beyond, ensuring progress and bringing hope. Today is a celebration of the class of 2025, a celebration of academic success, of persistence, of community, and of unimaginable possibilities. You have each traveled your own journey to reach this critical milestone in your career, often navigating uncertainty, obstacles, self-doubt, and making many sacrifices. But you have overcome these challenges, and in the process have learned more about what you are capable of and acquired life skills and values to make a meaningful difference in the world. And this is just the beginning. You are entering a world marked by complexity and uncertainty, but also one filled with immense opportunity for meaningful change. You carry with you not only knowledge, expertise, and skills, but also the values of this institution: science in service to humanity, integrity, curiosity, and a commitment to common good. Let your pursuit of excellence be shaped by purpose and sustainability for future generations. Let your innovation reflect empathy as much as ingenuity. And let your work be grounded in service, because when science is done in partnership with society for planetary health and human security, it becomes a force for hope for this and future generations. These are turbulent, volatile, and unprecedented times. But they are also times that call for the best in each of us, times for thoughtful, principled science, science that holds itself to high standards, dares to see the world differently, and chooses always to serve and unite rather than divide. Our hope lies in this ubiquity of science, on it transcending political and cultural barriers as a universal language to build bridges between the peoples of this world. Our interconnectedness and shared vulnerabilities underscore the importance of working together as scientists in service of humanity to generate knowledge and innovations to make the world a better place for everyone, everywhere. May we continue to learn from one another and move forward together. Thank you, Rockefeller University, for bestowing upon us your highest honor. Congratulations, class of 2025. I wish you strength, purpose, and joy as you continue on this wonderful journey of life and leave your own mark on the world. Thank you.
Vice Chair Steele, President Lifton, faculty, graduates, students, guests, it's a great pleasure and indeed an honor to be here with you this afternoon, joining the class of 2025. As you've already heard from our citation, we have been on a long road. We decided to focus on Africa's greatest challenge in the HIV epidemic, the high rates of HIV in teenage girls and young women. A challenge that took us to many different parts of understanding science, whether it was basic science in the laboratory, clinical research, social science research, all of those featured in how we needed to understand the problem. I hope to share with you in the next 2 minutes the 3 key lessons that I took away from this 40-year career. So it all started back in 1990 when we undertook this first study that enabled us to understand the nature of age-disparate sex, where teenage girls had the highest rates of HIV infection. Teenage girls had 8 times more HIV infection than their male counterparts. And we began to understand, and what we decided to do back in 1990 was to devote our research to solving that problem. How is it that we can slow down the rates of new infections in young women in Africa? That was the task we set ourselves. We made the first product where we worked with a company in the US. We supplied them with naloxone 9, and they made a film for us, and we did that first study back in 1993. What we found, naloxone 9 does not work. We undertook naloxone 9 studies with a gel formulation. We undertook studies of Pro 2000 gel, of buffer gel. We undertook studies over 18 years, all of them failures. When we went to an AIDS conference and we went up to the podium, people knew already that we were going to present yet another failure. Indeed, we became even known as experts in failure. We were that good at it. It took us 18 years of failure. For those of you trying to build a career, let me tell you, it's not a very easy way to climb the hierarchy of academia to keep failing. But it was in 2010 that we shared with the world our initial discovery that antiretroviral drugs can prevent sexual transmission. Today, something you all know as pre-exposure prophylaxis. It all started with a study of tenofovir gel. So what did we learn? What are the lessons from that experience that I can share with the class of 2025? Well, I'd like to share with you the 3 Ps. The first is find your passion. Find the thing that excites you to wake up in the morning. How else do you explain that after 18 years, we kept dealing with the same problem, we kept at it? We were not going to let it go. Why? Because the problem was too serious. Young girls having HIV presented a problem in that their whole future careers were at stake. They were going to pass this virus to their unborn children. We had a whole range of issues we had to deal with. We had to find our passion. So for each of you, I say to you, find your passion. Find the thing that excites you, that makes you want to undertake the next day. The second is that you've got to persevere. Life is full of obstacles. For those who decide to give up at the first obstacle, they will never achieve the kinds of important findings that make a difference in the world. You have got to find the passion that will enable you to persevere and to keep at it no matter what the odds against you are. And even when people call you experts in failure in the corridors. And finally, the last P, never compromise. The pursuit of excellence is the very heart of what enables all of us to move forward. And that pursuit of excellence is not a part-time activity. It's not something you do today and not tomorrow. For us, it has imbued everything we do, right down to how I organize my sock drawer. So, I leave you with this message, the class of 2025. Find your passion, persevere to achieve your goal, and may you pursue excellence in your future. Thank you very much.
Thank you both for those inspiring words. It's now my great pleasure to recognize the many wonderful contributions made by one of Rockefeller's most dedicated advocates and beloved friends, our life trustee, Marlene Hess. Marlene is a powerhouse in the philanthropic world and has an outsized impact on the field of biomedicine, as well as on education, culture, and the arts. But perhaps a better description of Marlene would be as a beacon, a woman who uses her remarkable intellect, empathy, and insight to make the world a better place. Deb and I have had the extraordinary pleasure of getting to know Marlene and have experienced firsthand her keen wit, deep integrity, and tireless concern for the needs of others. Our friendship with her and her husband, Jim Zirin, a brilliant legal mind who is equally adept with a pen and as an interviewer, and our relationship with them has meant so much to us in our time here at Rockefeller. Marlene's service as a Rockefeller trustee began more than 2 decades ago. However, her relationship with the university was sparked long before that. In 1980, Marlene's father, the legendary businessman and philanthropist Leon Hess, established an endowed chair at Rockefeller at the request of his friend and peer, David Rockefeller. That Leon Hess professorship is currently held by Rockefeller's celebrated cancer biologist, Ticha de Lange. There's perhaps no more prestigious introduction to Rockefeller than through David Rockefeller himself. In the intervening years, Marlene has demonstrated her dedication and commitment to this university and established herself as one of the university's most visionary benefactors and an unparalleled advocate for the institution. Marlene was elected to the Board of Trustees here in 2002 and elected as a vice chair in 2017. She's played an invaluable role on some of the board's most critically important committees, including the Executive Committee, the Compensation Committee, the Development Committee, Finance and Operations Committees, as well as the Educational Affairs Committee, which she chaired from 2013 to 2024. In 2024, to recognize Marlene's manifold contributions to the betterment of the university and the advancement of science, she was selected a life trustee of Rockefeller University. This is one of the highest honors Rockefeller can bestow upon a trustee. Marlene joined the Rockefeller board at a critical time in our history, when the university faced enormous challenges. We had a shortage of modern lab space, and our technology needs were growing exponentially. There was also a notable gap in women in leadership roles at the university, both on the board and on our faculty. Marlene's time as service as a trustee in dealing with these challenges spanned the tenures of 4 presidential administrations: Tom Sackmar, Paul Nurse, Mark Tessier-Levine, and myself. The changes that have occurred since the start of Marlene's tenure on the board are nothing short of extraordinary, and the achievements that have taken place are remarkable. Since 2022, 4 Rockefeller scientists have won Nobel Prizes. 2002, 4 Nobel scientists have won Nobel Prizes, 7 have won Lasker Awards, and 6 have won Breakthrough Prizes. Now, I won't say that those awards would not have been given had Marlene not been on our board, but I'm grateful we didn't have to do that experiment. At the same time, we've renovated over 1/3 of our lab space, created the Collaborative Research Center, and established new research cores to provide leading-edge technology and instrumentation. The Stavros Niarchos Foundation David Rockefeller River Campus was conceived and constructed, and the university established a translational research pipeline that's helping our scientists to move discoveries from bench into new therapeutics. With Marlene's leadership, the Women in Science Initiative has helped to showcase the work of women scientists and support their investigations. And this initiative has raised more than $50 million in funding for graduate and postdoctoral fellowships, the recruitment of women faculty, special research initiatives related to women's health, and women-led translational research projects. Today, approximately 40% of Rockefeller's scientists are women. In the same time period, the number of women on our board of trustees has more than doubled, and today there's an equal number of men and women on our board. Marlene's leadership has been instrumental in bringing about these advances at Rockefeller, and she has been equally influential in the field of biomedical science. It was Marlene who introduced Susan Solomon to Tom Sackmar, Rockefeller's acting president, in 2002. This fateful meeting led to Susan's founding the New York Stem Cell Foundation in 2005, and this organization has gone on to invest more than $450 million in stem cell science and has had an enormous impact on science and medicine. Now let me say a few words about Marlene's professional career and enormous pro bono involvements. Shortly after earning a degree from Mills College in California, she joined Chase Manhattan Bank in New York, where she served as Vice President of Corporate Communications and Manager of Public Services Marketing, eventually rising to the position of Director of Not-for-Profit Relations. During her tenure at Chase, Marlene played a key role in enhancing the bank's philanthropic activities. She created a range of campaigns to raise public awareness about community issues, including an award-winning child vaccination program that Chase spearheaded in partnership with the New York City Department of Health and the Children's Defense Fund. Marlene went on to serve as Managing Director of Global Philanthropy at J.P. Morgan Private Bank. She's advised clients from the U.S., Latin America, Europe, and Asia on topics as far-reaching as operations, governance, and outreach. In addition to forging partnerships between the corporate and nonprofit sectors, she designed innovative programs, conferences, and symposia for nonprofit organizations. Since her retirement from J.P. Morgan in 2005, Marlene has served as an independent philanthropic consultant, and many of the city's most distinguished organizations rely on her leadership and expertise. When I came to Rockefeller, I thought it was remarkable how great our board of trustees were and how incredibly generous they were with their time and philanthropy with Rockefeller. And I thought it's really remarkable that all the great institutions around New York must have their own set of trustees who are equally generous. And what I learned rather quickly, that it's not actually a multiplicative set. It's actually the same set of people who play incredible roles for almost every imaginable New York center of excellence. And Marlene is a good exemplar of this. Marlene is also vice chair of MoMA, treasurer of the Lincoln Center, and has served in leadership roles at the New York Presbyterian Hospital, the Metropolitan Opera, the American Museum of Natural History, and the New York Stem Cell Foundation. Now, I don't know if this is an indoor world record or not, but we can count them up later, Marlene. Marlene is also an active member of the Council on Foreign Relations and has served on a variety of its key task forces. She was presented with the Visionary Leadership Award by the International Women's Health Coalition in 2019, and in 2022 was honored as a living landmark by the New York Landmarks Conservancy. Marlene's philanthropic contributions have been many and varied. Her impact at Rockefeller has been nothing short of transformational. Continuing in her father's footsteps and simultaneously paying tribute to him, in 2006, Marlene established a second Leon Hess Professorship, which is now held by cancer biologist Sohail Tavazoui, who you've heard from several times today. In 2016, her philanthropy helped to create a new pavilion on the Stavros Niarchos David Rockefeller River campus. The Hess Academic Center, dedicated to her father's memory, is home to the President's Office and the Dean's Office. It's a privilege for me, and I know it is as well for Tim Stearns, to go to work each day in a building bearing the Hess family name. Most recently, and in a truly visionary act, the Hess Philanthropic Fund has established the Marlene Hess Center for Research on Women's Health and Biomedicine at Rockefeller. There is no other basic science institution with a major initiative devoted to studying human health through the lens of sex differences. We do not yet understand why certain diseases and disorders affect women and men differently, which is why it is so important to try to understand these differences at their biological root so we can develop improved approaches to therapies. The Hess Center is led by physician-scientist Agata Smorgozewska, who you've also heard from today, who heads the Laboratory of Genome Maintenance. Agata is herself an alumna of Rockefeller University and brings an extraordinary level of experience and caliber of research to this new role. Marlene's philanthropy has been truly remarkable at Rockefeller, but such commitment is only possible because she has an innate understanding of the importance of the work underway here and of our unique and remarkable culture. She knows that the university is its own community, and its sense of inclusiveness must be cultivated and protected. A hallmark of Marlene's trustee service is extending the warmth of the community to every newcomer. In this way, she has sustained David Rockefeller's spirit of excellence and civility. Marlene has dedicated so much of her time and energy to Rockefeller, and we are grateful to Jim, her children Peter, Maggie, Ann, and David, and their families for sharing her with us. Marlene, congratulations, and thank you so much. Congratulations, Marlene. All right, I'll give you that. Podium is yours, and this is yours too.
I don't know who she is, but I'd like to meet her. Thank you, Rick. It was very beautiful, and I'm very touched. It was a very generous tribute. Thank you, Bob Steele, sitting in for Bill Ford. You're wonderful to be doing this. Thank you, fellow trustees and distinguished faculty. I'm sorry to have my back to all of you. I didn't really understand that that was going to happen. I'm deeply proud to be part of Rockefeller. It's been a great pleasure to serve as a vice chair and as a trustee. I'm really thrilled to be receiving this honorary degree today. I'd also like to thank many of you with whom I've worked over the years. Mike Young, my fellow seatmate, Corey Bargman, let's see where everybody else is. Barry Kohler, I think you're over there. Tim Stearns, Sid Strickland, who I haven't seen, but he may be here, and Marnie Imhoff. Also three who, as you heard, almost count as family. The two Leon Hess professors, Tizia Delanga and Sohail Tavazoy, and Agata Smogojevska. I did it, didn't I? Agata Smogojevska, director of the Hess Center for Research on Women's Health and Biomedicine. I'd like to congratulate all of you, class of 2025. I'm so happy to be here with you to celebrate your really sincerely great achievements. I've enjoyed meeting many of you at Educational Affairs Committee meetings and have been fascinated by your research presentations. I must say I am deeply humbled to be in a class of honorary degree recipients that includes such amazingly great scientists. It is just thrilling for me to be here. When thinking about it today, it occurred to me that the Rockefeller University campus is just across the river from Four Freedoms Park. Many of you know that. It's the memorial celebrating Franklin Roosevelt's 1920s vision. President Roosevelt outlined 4 freedoms that he thought were essential to a just and peaceful world. They are freedom of speech and expression, freedom of every person to worship God in his or her own way, except they never said it like that, freedom from want, and freedom from fear. But it seems to me there's at least one more crucially important freedom, and that is the freedom to explore, investigate, and research the fundamentals of what life is all about. Rockefeller's mission, as you've heard today already, is science for the benefit of humanity. And Rockefeller is home, as you know well, to some of the most innovative scientists producing truly transformative research discoveries. The scientific freedom and collaboration here that takes place is absolutely thrilling. This could not have been possible without the Rick Lifton and the extraordinary leadership, Rockefeller leadership over the years. And I actually thought there were 6 Nobel Prize winners while I was here. And they are Gunter Blobel, Paul Greengard, Rod McKinnon, Ralph Steinman, Mike Young, and Charlie Rice. During this time of chaos, when so many of our fundamental beliefs, rights, and protections are challenged, I am truly heartened to think of the magnificent science that's going on here. How lucky we are that Rockefeller University is in our lives. I'm deeply grateful to all of you remarkable scientists of the class of 2025 who will be going forward to advance crucial freedom to investigate, research, and explore science for the benefit of humanity. Humanity needs all of you more now than ever. I can't tell you how much it means to me to be with all of you today receiving Rockefeller's honorary degree. Thank you so very much.
Technology remains elusive. Well, now, for our final honoree today, it's my great pleasure and honor to introduce Rockefeller's Richard and Jean Fisher Professor and Head of the Laboratory of Genetics, Michael Warren Young. As a youngster in southern Florida, Mike relished his treks into the rock pit, an area on the edge of his neighborhood. There, he climbed hills among skittering lizards and slithering snakes. He poked around in ponds and caught darting tadpoles, which he brought home, hoping to watch them turn into frogs. These explorations, however, came with some peril. A no trespassing sign at the rock pit included the warning, "Violators will be prosecuted." And Mike thought, at age 9, that prosecuted meant electrocuted. Yet this did not deter him. His willingness to take risks to serve his adventurous spirit wove through his life and into crucial career decisions. Mike's naturalist sensibilities expanded into other subjects. During his senior year at the University of Texas, Austin, Mike took a genetics course with Burke Judd, a noted fruit fly geneticist. Mike impressed Judd with his incisive questions, and Judd invited this curious student to join his lab for summer research. The experience captivated Mike, and he stayed on for graduate school. Judd's group was probing conundrums that were emerging from the study of fruit fly chromosomes. For example, the fly genome contained enough DNA to encode about 20 times the number of genes that scientists had identified. Some were vital for survival, and others controlled conspicuous morphological features. What was the rest of the DNA doing? Mike developed a new method for inducing chromosome breaks, creating flies with interruptions in their DNA. Investigators could then check whether any of these mutations produced traits of interest, not just those that were essential for life or dictated, for example, eye color. For starters, Mike tested whether the disruptions that he generated in the X chromosome compromised fertility or other traits. In 1971, while Mike was conducting this work, researchers at Caltech, Ron Konopka and Seymour Benzer, discovered strains of fruit flies whose usual 24-hour clock, their circadian rhythm that couples activity to the day-night cycle resulting from the Earth's revolution on its axis, was absent, producing flies who had chaotic activity throughout the 24-hour day rather than the normal 12-hour period of high activity followed by 12 hours of markedly reduced activity. Fortuitously, the responsible gene, called the period or PER gene, resided in the region of the X chromosome that Mike was studying. And he found that one of his fly strains contained a break in the period gene, disrupting the normal circadian rhythm. By the time he finished his Ph.D. in 1975, the recombinant DNA revolution was exploding. Mike moved to the Stanford Biochemistry Department to do postdoctoral research with David Hogness. There, he dove into the new techniques of molecular genetics. The Hogness lab had been the first to foresee that recombinant DNA technology, conceived by a graduate student in the department, would ultimately allow the cloning of genes and mutations that cause any inherited trait in the fly, and eventually in humans. 3 years later, he was ready to launch his own lab. Rockefeller appealed to him, although a position here came with risks. The promotion process was regarded as brutal and opaque. Tenure was elusive. But the institution offered immense benefits. He'd be surrounded by terrifically accomplished colleagues, and without undergraduate teaching responsibilities, he could focus on research. I also remember him coming back and telling me that they told him he could have a parking space, and that was considered a perk. Something for a fellow who drove a Norton motorcycle did not have the immediate appeal that people at Rockefeller thought it would. Nonetheless, at Rockefeller, he decided to come and take on the risky project of trying to track down and capture the DNA segment that contained the PER gene first identified by Konopka and Benzer. No one had previously defined a gene that controls behavior, and conventional wisdom held that doing so would be impossible for such a complex trait. But Mike knew that he possessed a mighty asset, a break in the period gene showing that a single mutation was capable of producing this phenotype. And so he used his time and freedom offered by Rockefeller to pursue this very high-risk project. In 1984, Mike reported isolation of the DNA segment encoding the period protein. Next, he wanted to know whether on its own it would restore normal circadian rhythms to flies that carried flawed versions of this gene. Mike was able to introduce the normal period gene back into those mutant flies, and then recorded the activity of flies over multiple 24-hour cycles. The team could thus see whether the activity patterns reverted from chaotic behavior to regular, recurring 24-hour cycles. After several days, the answer became clear. Adding back the period gene restored normal circadian rhythm. The researchers reported this discovery at the end of 1984 with similar results published by Mike Rosbash and Jeff Hall at Brandeis. Mike had his hands on a single gene that controlled behavior, but magnificent as this achievement was, it did not immediately illuminate mechanistic details of how this clock worked. Mike's lab then did a heroic genetic screen, introducing random mutations into flies and then screening them one by one for defects in circadian rhythms. This large-scale screen identified a mutation in a different gene, which they named timeless, that also caused loss of normal circadian rhythm. They went on to show that the timeless protein physically associated with the period protein. Further studies showed, and this of course was very critical because it suggested that these two genes were operating in the same pathway and that there weren't multiple independent ways of regulating circadian rhythm in the fly. Further studies showed that the PER gene expression begins early in the day and that the PER protein accumulates in the cytoplasm. The timeless protein begins accumulating later in the day, binds to PER, and when its level gets high enough, typically in the evening, this complex enters the cell nucleus and turns off expression of the PER gene, establishing an on-off cycle for the controlled expression of PER in sync with the 24-hour day-night cycle. They went on to identify light-sensitive proteins in the eye called cryptochromes that make the cycle light-sensitive by altering the timing of production of another protein Young's lab discovered called double time. The beauty of this mechanism is that it allows the sleep-wake cycle to adapt to changes in when the sun comes up and goes down, as occurs with changing season, and also when we travel to different time zones. Mike's lab, along with now many others, has gone on to show that circadian clock genes are present not just in flies, but across the animal kingdom. And humans with defective versions of these genes experience perturbed sleep-wake patterns as well. For example, Mike and colleagues at Weill Cornell Medicine and Bilkent University in Turkey associated a particular variant of a clock gene that underlies chronic sleep perturbations in a sizable portion of the human population. In addition to regulation of the sleep-wake cycle, these circadian clocks operate in individual cells throughout the body. These findings have important implications for not just jet lag with travel, but for understanding the challenges from changes in work schedules among shift workers and medical residents that make sleep schedules chaotic. In addition to running his brilliant research program, Mike has made extraordinary contributions to the Rockefeller University as Vice President for Academic Affairs from 2004 to 2023. Prior to 2004, Rockefeller was often a hierarchical institution in which one could only become a professor by having been a junior faculty member under the leadership of a more senior professor. This organization made talented junior faculty wait for years to have the chance to pursue their own original ideas. This had evolved to, in some cases, result in small committees being convened to conduct targeted searches for scientists in particular disciplines, which resulted in new hires, but not always the best people in the field. Mike and then President Paul Nurse decided to change course and in 2004 instituted an open search process, seeking the most promising and exciting scientists in any field and recruiting them to Rockefeller as independent investigators to pursue truly audacious goals. Now, Rockefeller's open searches include about a third of the faculty to collaborate and identify those scientists whose work has the potential to make transformational contributions to their fields of inquiry. Furthermore, the process of gaining tenure has gained transparency, and new investigators know the university is deeply invested in their success. The current strength of our faculty is directly attributable to the 29 faculty who have been recruited since 2004 in the open search process. Mike has done a tremendous job in his 20 years as Vice President for Academic Affairs. Mike's research, which provided the first molecular mechanism of how a changing environment a changing environmental cue, for example, the timing of sunrise and sunset, induces a change in behavior such as the sleep-wake cycle. Suffice to say, this was an enormous advance that has been well recognized for its importance. Mike is an elected member of the U.S. National Academy of Sciences and the American Philosophical Society. He's also an honorary member of the UK's Physiological Society. He's received many awards for his pioneering research, including the Louisa Gross Horwitz Prize, Canada's Gairdner International Award, and the Shaw Prize in Life Sciences and Medicine. But I most fondly remember waking up on October 2nd, 2017, just prior to 5:30 a.m., perfect time to turn on the television and see the broadcast from Stockholm of the announcement of the Nobel Prize in Physiology and Medicine, which named Mike and Michael Rosbash and Jeff Hall as recipients, an extremely well-deserved recognition. Many of us on campus that day commented on how rare it was to have someone who was so congenial and so graceful and so beloved by his peers be so distinguished as a scientist and so effective as an institutional leader. Mike, with great admiration, please come up and get to it. Congratulations, Mike. Here, you take a hold of it.
Podium's yours, buddy. Thank you. I'll leave this with you.
So first I want to thank the university for bestowing this honor. It was a surprise when I got the message, but it's a deep honor. I've had a tremendous amount of luck in my career, and I think we've talked about a lot of failures and worrying about failures, but it's also important to think about accidents that happen as you're doing your research. Accidents, I think, are very commonly linked to discoveries. I mean, if you knew it already, if there wasn't an accident, how could it be a discovery? And I think my career has been full of those kinds of issues. And in fact, the path I've described reminds me of a book by Stephen Gould, Wonderful Life. Gould was a paleontologist. He was fascinated with the array of organisms living 500 million years ago, and he argued that many fewer branches of animal life are present now than at that time. Although from the abundance of all of those fossils, all of them seem to be very well adapted 500 million years ago. So he points to the likelihood of accidents of time and place, and argues that rolling the evolutionary dice again might produce a very different world than we have today. The title of his book was swiped from an old black and white movie. I hope many of you are familiar with it. It's called It's a Wonderful Life. It plays just about every year around the winter holidays. It's about George Bailey, homegrown pride of his town and supremely honest banker that makes a mistake that spells misery for his friends and his family. He wishes he'd never been born, but an angel takes him to see what would have happened to the town if that were true. His brother is missing. The angel points out that George saved his kid brother from drowning, so no George, no brother. And of course, George's kids are also missing. His wife never married, and she lives stoically in an old house that is falling apart. So you've heard what Rick said. I think I have much in common with George, a randomly chosen course in genetics. Something that I think Rick didn't point out is that at the very beginning, I was a senior in college in 1971. I needed a course in genetics in those days at the University of Texas. You'd go to this enormous gymnasium and you'd come in with an index card and you'd fill out your courses on the index card. And I went up to a table for biology and I said, I need a genetics course on a particular day of the week at a particular time of day. And this is what they gave me. And that turned out to be Burke Judd. And Burke had asked, as Rick mentioned, Burke asked if I'd like to do a summer of research. I said, yeah. And that started this ball rolling. So again, I think I have much in common with George Bailey in that old black and white movie, a randomly chosen course in genetics, a project as a graduate student that set me up to find a clock gene, but was initially pursued for a very different purpose. And recombinant DNA just happened along right at the time when the transition from an almost Stone Age form of genetics to a wondrous form of molecular biology had been invented. And the timing of all of these things, if they'd been spread out, it wouldn't have happened. And so, again, I refer to this nature of accidents of time and place as being incredibly important. I think the appeal of the old movie may be its broad relevance and a fairly simple theme. Accidents of time and place are bound to happen. Just hope you've made the best of whatever has crossed your path. Finally, I want to, in addition to thanking the university for this honor, I want to thank them for 47 years of supporting my research. It's been an extraordinary place to work, extraordinary colleagues. Its unusual structure makes it incredibly powerful, and of course the history shows that. And finally, I want to congratulate all of you students who've come from this extraordinary university on your graduation. It's been hopefully you'll have many accidents like I did and be able to take advantage of some of them. Thanks again very much.
Thank you, Mike.
So, Quarraisha Abdool Karim, Salim Abdool Karim, Marlene Hess, and Mike Young, please rise. By the authority of the Regents of the University of the State of New York, which they have invested in the trustees of the Rockefeller University and is by them delegated to me, I confer upon you the degree of Doctor of Philosophy, which admits you to all the rights and privileges thereunto pertaining. Congratulations. Let's see. So we're now entering our seventh, no, that's okay. Okay. Honorary, you have been a marvelous crowd. Honorary degree recipients, Vice Chairman Bob Steele, graduate students, faculty, staff, students, and friends, and graduates. This concludes the 67th Convocation of the Rockefeller University. On behalf of the graduates and honorands, I'd like to invite you all to a reception on the lawn outside Abbey Aldrich Rockefeller Hall immediately after we leave. So, please remain at your seats until the marchers have exited the auditorium. Once outside the auditorium, the graduates and honorary degree recipients, along with everyone else on the stage and the faculty presenters, will assemble for a class photograph on the steps of Rockefeller University Hospital. So following the photograph, the graduates will join their families and friends at the reception. And now I ask that you all please rise.